He was born in Barranquitas, Puerto Rico. He holds a bachelor's degree in chemistry and biology, a master's and a doctoral degree in biochemistry. He has worked as a faculty and researcher in Puerto Rico, the United States, and other places in Latin America. He is a professor in the areas of chemistry and biotechnology. He also served as academic dean and currently holds the chancellor position at Barranquitas campus. His research focuses on biotechnology applications in agricultural crops, the mechanisms of actions of heavy metals in animals and plants, the epigenetics of cancer. He has developed undergraduate and graduate academic programs, including the first doctorate degree in biotechnology in Puerto Rico. Through proposals from public and private funds, he has developed several projects, such as the Institute of Sustainable Biotechnology, which is a research and learning center at the Interamericana campus in Barranquitas. Agroevolución Puerto Rico, which provides training and in vitro plants to farmers, and the Center for Food Security and Sustainable Agriculture, which is expected to open this year. We welcome Dr. Juana Negrón.
Can you hear me? Yeah? Okay. So, hope you got lunch. And I hope you don't get sleepy. Okay. So, probably this is one of the most recognized pictures of the Caribbean. Ahora. That was better. So probably this is one of the best pictures of recognized places in the Caribbean. They have sea, sun, good weather, and many other things. But there are other things in the Caribbean, and food and agriculture is very important. I will explain a little bit about agriculture in the Caribbean. I will talk about sustainable agriculture in Puerto Rico and the possibility for improving our way of life. This is the Caribbean. Puerto Rico is, we consider Puerto Rico a very small island. There are many other islands in the Caribbean, and usually when we talk about the Caribbean, we refer to those places which are close to the Caribbean Sea. So in that sense, Caribbean could be the northern part of Venezuela, Colombia, and other places. However, in this presentation, we will concentrate maybe in the islands of the Caribbean, including, of course, Puerto Rico. This is the population of the Caribbean. These are the main islands that we will consider in the presentation. And this data was presented by Dr. Sonia Peters a while ago. The history of agriculture in the Caribbean. It has been linked to exportation, the colonial times. That means that in the past, like other speakers mentioned today, in the Caribbean, we used to produce a lot of food. Actually, we were exporters from Europe and many USA and many other parts of the world. The main problem with that is that the capital was not from the islands. So it was a foreign capital, and of course, the interest of that market and that agricultural activity was not directly to serve Caribbean countries, okay? And even worse, today, agriculture is only a small share in the economy of all those islands. Of course, there are some exception, for example, Dominican Republic. It has a big share of the economy compared to the other islands in agriculture. Puerto Rico is an example of this history of agriculture in the Caribbean. One of the characteristics of that agriculture, probably in the 19th, 18th century, was an agriculture concentrated in monocultures. For example, here in Puerto Rico, in one time, everybody was planting tobacco. Later, sugar cane. The problem with that is that first, the capital is not a national capital, and second, that many other important crops are being abandoned. And many other important crops like coffee, rice, for example, in Puerto Rico, you saw it in the lunch. Our main dish is what? Rice and beans. And we don't produce none of them. We should be eating other kind of food, because that means if there is a problem with the transportation with the rice and beans in Puerto Rico, like in Maria, what are we going to do? So this displacement of crops is one of the biggest problems in the Caribbean. And of course, like other of the speaker mentioned, agriculture has been abandoned throughout the years. For example, in Puerto Rico, we had, for example, sugar cane, we had tobacco, and one time we had coffee. Even here we said in Puerto Rico that the Puerto Rican coffee is so good that the Pope used to drink that coffee. I don't know if that's true, but certainly we have a good coffee. But if you look at the numbers, even there is, if you want to import coffee to Puerto Rico, there is a tax. There's some sort of protection to our coffee. But do we produce all the coffee that we drink? No. We are not close to. So, and later, the other change in the economy in Puerto Rico was the heavy industrialization. For example, those with the hair a little white like mine, remember what we used to have between Ponce and Peñuelas, la petrochemical industries. We don't produce oil. And what they left? Contamination, disease. That's what the petrochemical industry left in the island. This is the gross domestic product of many countries in the Caribbean in comparison with the amount of dollars in each one of those countries. You can see, well, in general, you can see that the gross domestic product is very low in most of the islands. There are some exceptions, like I mentioned. For example, Dominican Republic, you see Guyana in South America. But most of the islands are in this part, including, actually we will need probably a lower scale for Puerto Rico. How much, well, what is the contribution of the agriculture in Puerto Rico? I think Terry mentioned 1%. It's less than 1%. This is the reality, according to the Food and Agriculture Organization, of food insecurity in the Caribbean. When you compare with the rest of the world, the orange bar, quite high, okay? We are talking about over 60% of food insecurity. For example, in terms of where do we get the product that we consume, mainly from USA, but there are other places like Europe. What it means is that we are in a pretty bad situation regarding the food that we consume every day. You can see here the amount of importation in billions of dollars. That means that trend keep on growing. So that means that we, Caribbean region, have to do something to change that. When we talk about food security, what we are talking about is the amount of food that we can get, the quality of that food. I mean, what kind of food are we getting in our tables every day? Do we have access to that food every time, no matter what happens? For example, in Maria, here from this campus, we have the only supermarket here in Barranquitas is to that direction. And you can see it from here. And I remember seeing all those lines of people waiting for a pack and probably a couple of pounds of rice and beans because we don't produce anything. And even the most frequent food that we consume is not produced in the island. And of course, food security had to do also with culture. Okay? A product that we consume in the island, that we produce in the island, is very good for the community because it means that it creates employment. However, that food security can be linked to nutrition. And this morning, that idea was mentioned. For example, we could have nutrition insecurity even if we get big amount of food, but what is the quality of that food? Okay? It have to be safe to the environment, and also we have to start to implement different practice in order to change that. For example, if you have an unhealthy diet, these are the main consequences. For example, during certain development stages, we need certain kind of food. If you don't take that food, you can have problem with the development. You could have anemia, and probably one of the biggest risks, and what is happening right now, is obesity, overweight and obesity. Because that means that, for example, here talking about, for example, in Puerto Rico, we eat a lot of starch, a lot of carbohydrate, but we eat a lot of fat, a lot of saturated fats, okay? So that means that we are exposed, actually in the island, cardiovascular disease is the second cause of death. Why? Because the way we act, the way we carry out our life, we don't do too much exercise, but also on what we eat, the kind of food that we eat. So you are exposed to cardiovascular diseases, and you're also exposed to other things like diabetes. This is a summary from the stages of obesity in the Caribbean, way above the rest of the world. So we tend to have more mass, but not good mass. Lipids, fun, okay? And the problem with this is in Puerto Rico there have been made many studies in the past, and they have shown that this is worse in the kids, in the children. I mean, we adults, all of us, no, not all of us, most of us have many pounds over what we should have. But in the kids, especially elementary school and high school, the problem with obesity is really dangerous here in the island. And this is the kind of thing that we import. Not only the food to be prepared at home, but even in the restaurant. How many calories has a double meat hamburger with bacon and all that? Probably the calories that you need for the whole day. For the whole day. He said, what do you want? Water or soda? No, I want soda. There you have probably 500 more calories. And there you have a lot of compounds that are not healthy. A lot of fructose, for example, which has been linked to cardiovascular disease. Okay? There are many other bad consequences for bad nutrition, the obesity, but the thing is that most of these products that we import, they are high calorie content. Even Puerto Rico, we are in a worse position. When you go to look for the data of Puerto Rico, what it says, no results available to Puerto Rico. So that means that we don't pay attention to that. The academic, there are not too many studies in this area in Puerto Rico. The government, the common citizens don't pay attention to that. Because we think we are an island and we have everything we need here. Okay? And that's not true. If we want to improve our agriculture, if we want to improve our resilience, if we want to improve the quality of our food, one of the important things to do is to generate the data and share that data to be part of the world. So this is very unfortunate. Puerto Rico government does not share the data. It's not available. Of course, there are some studies of Puerto Ricans. Where do we have the majority of Puerto Ricans? USA. So this is a study from Hispanic Puerto Ricans in USA. But we are not represented there. Even in the USA, our food insecurity in USA is not good compared with other communities in that nation. This is a comparison within the Hispanic community. If you compare that with other cultures, it's worse, the difference. There was a study made by a couple of fine scientists from the Universidad of Puerto Rico and Rio Piedras, and they identified several variables to try to estimate the food security in the island, okay? And if you see, as more greener, the situation is worse. So can you see how many areas do you see clear of low risk? Not many. Probably the worst situation are the municipalities in the interior of the island, where many times you find the poorest people in Puerto Rico. Do you think agriculture can make a difference? Of course. Not only to have more farmers growing food for everybody, but even at our homes, like you were mentioning. You see, this data, I think it's 20, that was previous to Maria. So that means, what is the representation of agriculture in the economy of Puerto Rico? 0.8%, less than 1%. So Puerto Rico is in a high risk, absent of agriculture, productivity, treat Puerto Rican food security. You can see here the impact, not only the history, natural phenomena, hurricanes, climate change. We expect more hurricanes due to the climate change. So that means that we are more in risk in this moment. Puerto Rico produced, and I will use a number which is probably worse, only 50% of what we eat. If you go to the supermarket and you have $100, $85 will be invested in importation, okay? Very low variety of local product in the supermarket, and many times it's not consistent. You can find it certain month of the year and all that. I think Terry Hans mentioned something similar to that. These are the farms dedicated for food production in Puerto Rico. Every year we have less land being used for cultivation, for the production of food. You mentioned then the solar panels, okay? There are wonderful lands that have been used for solar panels. I'm in favor of solar energy. In this campus, 25% of the energy comes from solar panels. But there are places to localize these panels. We can use the roof of the houses. But we use, let's say, somebody decides to use El Valle de Lajas. It's one of the best places in the island, in the southern part of Puerto Rico. I believe in solar energy. But I think we should go to the homes, to the roof of each one of the houses in the island. There are many other problems with food insecurity in Puerto Rico. We have the Jones Act. That means that a merchant marine rule requires that all imports from sea be processed through United States. But we see the advertisement in the TV. They are here for us. That's what they say. We are here for us. I mean, to bring you happy. No, they're charging a lot of money in the transportation. That means that our pocket will pay that. Okay? In addition to that, like I mentioned before, the weather. You know, many of us have seen what was the result of Maria. Plantains, chickens, all the product went away during those days. Conventional agriculture. So what kind of agriculture do we want? The conventional agriculture requires a large capital investment. It concentrates in monocultures. It uses large-scale farm. It requires a lot of mechanization, pesticides, herbicides, and fertilizer. And this is the model that prevails in the world. But here in Barranquitas, that's the model that we can adapt. I don't think we have the topography, there are many other things that are not good for that model. So we should go for an ecological alternative to avoid what? In addition to this problem, we have problem with the water, contamination, habitat, the loss of biodiversity, carbon emission, and many other bad things that not only creates problem with the crops, but also creates a problem with the climate change. So we consider that you should have ecological alternative, but also high technology. For example, you see this, how this building is called. It's called sustainable biotechnology. Two terms that many times you don't associate that they can work together. And there are possibilities to use biotechnology in a sustainable way. This some of the result of Maria. It was this picture is very common here in Barranquitas, Bonito, and many other municipalities close to the campus. Okay? So it means the Caribbean agriculture must take into consideration, like I said, technology, sustainable biotechnology, the market. We have to look at the consumer expectation and the weather, okay? Agricultural biotechnology in the Caribbean. That means that areas like biotechnology offer different tools to improve our agriculture in a sustainable way. For example, this morning we were talking about genetic engineering crops. I believe in sustainability. I believe in ecological management of the land, but I also believe in biotechnology. How many people have died because they have eaten a transgenic crop? Do you know somebody? No? So we have to work with the community. We academics have the responsibility to talk to the community and to explain. Also give participation to the community in this process. This is very important. That's why I admire the work that Dr. Peters is doing in Barbados. Precision agriculture. For example, here in the mountains, precision agriculture has many alternatives. You can do remote sensing. Here you can measure chlorophyll content. You can do many things using technology adapted to the topography of the land. We can reduce the amount of land that we use. Environmental control agriculture has been developed, have many developments that we can take advantage of. And adapt that to the climate change. We will create more jobs, and we expect prosperity to everybody in the community. This is an example of an alternative tissue culture. We provide tissue culture to the farmers. Initially, it was difficult because they said, well, how is this? A plant in a tissue culture will produce plantain. But for my surprise, they accepted quickly. And we had more than 100 farmers working in this project. Of course, at the beginning they were insecure and a little bit scared. This is an institute. We do teaching, research, and extension. We work with farmers and other members of the community. And these are some of the options for the Caribbean agriculture. First, we have to give the priority that it really needs. I mean, if you don't put it in your priorities, I say government, community, and all that, we will not do anything. We should implement climate-smart agriculture practices, partnership, collaboration within the Caribbean region, because it's our problem. We have to solve it. Nobody will come from outside to solve our problem. We need a strong government commitment and policies with the new agriculture. Of course, private groups should make a strategic investment in this area. Capable institutions like universities, like research centers, the community should be involved in this process. And of course, we should look to eat more healthier product and alternative. Muchas gracias.
Thanks, Dr. Negron, for your presentation. We have, do we have questions? Well, then that means that you explained everything perfectly.
Or they did not understand. I don't know. Thank you.
Thank you. Now we will have Dr. Jaime Sanchez. He was born in Bogotá, Colombia. In 1986, he obtained his bachelor's in agronomy from the Universidad Nacional de Colombia at Palmira, under the advice of Dr. José Salinas. In 1989, he obtained his master's degree del Centro Agronómico Tropical de Investigación en Turrialba, Costa Rica, under the supervision of Dr. Donald Glass. From 1989 to 1993, he was associate researcher at the International Center for Tropical Agriculture at Palmira, Colombia, in the Cassava program, under the direction of Raúl Moreno. From 1993 to 2000, he worked at Nestlé Research and Development, located at Quito, Ecuador, where he was elected head of the agronomy department in 1998. In 2004, he obtained his PhD on soil and water science of the University of Florida under the direction of Raúl Milavarapu, if I said that correctly. Since 2004, he has been at Salinas Research Center leader of Pioneer Hybrid Puerto Rico, and from April 2017, a Corteva leader in Puerto Rico.
Buenas tardes a todos. Hoy voy a presentar, pensando en la idea del seminario, pensé que tenía más sentido hablar sobre las metas de sostenibilidad de Corteva. Pero oyendo la presentación del doctor Negroni, me excusan por no haber estado hoy por la mañana en las presentaciones, me doy cuenta que también habría sido valioso tal vez adicionar alguna información sobre las compañías semilleras que están en Puerto Rico. Y voy a hacer una rápida revisión sobre eso. Todos creo saben ahora que la mayor parte o las compañías semilleras más importantes del mundo están colocados aquí en Puerto Rico. Y es una decisión que no ha sido tomada en los últimos años. Las compañías semilleras se empezaron a establecer en Puerto Rico a mediados de los años 80. Y no es curioso, es sencillamente porque Puerto Rico permite sembrar los cultivos de interés de las compañías semilleras norteamericanas pues casi que cualquier semana de las 52 semanas del año, ¿correcto? Entonces tiene sentido, ¿no? Uno puede hacer avances genéticos más rápido. Si uno puede sembrar 2, 3, 4 veces o más cultivos secuenciales en un año. Las compañías semilleras ahora son parte importante, digamos, de la operación agrícola puertorriqueña. Son empleadores, yo creo que son los empleadores en este momento, los empleadores número 1 de agrónomos de Puerto Rico. Pero también empleamos un montón de otras personas que tienen grados relacionados con la agricultura, incluyendo estudiantes que han estudiado en la Interamericana de Barranquitas o la Interamericana de Guayama. La gestión de estas compañías está relacionada, por supuesto, con la producción de semillas, ¿no es cierto? Producción de nuevos materiales de semillas. Y, por supuesto, el compromiso de los empleados, el primer compromiso de los empleados, pues es lograr las metas de las compañías, ¿no es cierto? Pero hay también una responsabilidad supremamente importante de todos los empleados de la compañía que tienen que ver con el valor de las compañías en la sociedad. Somos parte integral de la sociedad y hacemos contribuciones no solamente a la compañía, pero también a la sociedad. Entonces mi presentación hoy va a estar relacionada con la sostenibilidad, cómo enfrenta Corteva el problema de sostenibilidad y además cómo enfrenta los retos relacionados con el medio ambiente y con la sociedad, o sea, cuáles son nuestras metas en términos de medio ambiente y sociedad. Este es un statement general porque estamos diciendo aquí algunas cosas relacionadas con nuestra operación que pueden modificar la percepción sobre las acciones, pero espero que no haya aquí inversionistas. Muy bien. ¿Cuál es el tema importante? Bueno, todo el mundo sabe, estamos experimentando un aumento de la población mundial, ¿no es cierto? Para 2030 se espera que seamos 8.6 billones, para el 2050 9.8 y para el 2100 11.2 billones. Y obviamente la producción de cultivos es una cosa supremamente importante para poder abastecer, digamos, a esa población creciente. Pero no es que no se haya hecho nada, ¿no es cierto? La realidad es que los rendimientos globales de cultivos han aumentado un 30% en los últimos 20 años y eso hace que podamos producir más productos agrícolas en menor área. Entonces, desde ese punto de vista, ¿no es cierto?, la investigación en la producción de rendimientos más altos tiene que ver con la sostenibilidad de un mundo con una población creciente. Ese aumento de rendimiento ha prevenido que 240 millones de acres de tierra, de hectáreas de tierra, hayan sido utilizadas en la producción de cultivos. Es decir, para obtener el mismo rendimiento habría que haber usado 240 millones si los rendimientos no se hubieran incrementado. De manera que pues, es claro, es obvio que la agricultura está llamada de nuevo, ¿no es cierto?, a satisfacer las necesidades de esa población. ¿Cuál es la visión de Corteva? Por un lado, reconocemos el crecimiento de la población y lo que quiere decir que eventualmente los recursos naturales van a estar sometidos a mayor presión. Por el otro lado, reconocemos que la ciencia y la tecnología, como dijo el doctor Negrón, son un eje fundamental para producir alimento y combustibles en el mundo. Y por el otro lado, creemos que las empresas que trabajan con tecnología tienen la posibilidad de desarrollar aún nueva tecnología, o sea, que la tecnología que se ha desarrollado ha logrado los incrementos de rendimiento, pero tenemos además más posibilidades para desarrollar mayor rendimiento. Nuestras metas en términos de sostenibilidad y de medio ambiente y sociales están entonces relacionadas con 4 áreas fundamentales: innovación sostenible, biodiversidad, clima y, por supuesto, inclusión, diversidad y equidad. Sobre los 4 puntos vamos a hablar un poco más en detalle. Sobre innovación sostenible, estamos comprometidos a crear un pipeline de soluciones que sean, que logren satisfacer nuestros criterios de sostenibilidad para el 2025. En biodiversidad estamos preparándonos para asegurar que 25 millones de acres en biomas diferentes pueden recibir los productos que son amigables con el ambiente y con esos biomas. En el clima estamos tomando, claro, porque en el clima la influencia que puede tomar sobre el clima es sobre la operación misma de la empresa, ¿no es cierto? Entonces, en el clima estamos hablando de las emisiones que produce la compañía, cómo podemos controlar las emisiones, cómo podemos reducirlas. Y en el último lado, ¿no es cierto?, está el asunto de inclusión, diversidad y equidad, que está relacionado con el incremento de mujeres en posiciones de dirigencia de estas compañías, también en la inclusión de personas con diversidad de origen, educación. Todos sabemos que la diversidad es la fuente inagotable de creatividad y la equidad, ¿no es cierto? O sea, a todo el mundo debería pagársele de acuerdo a su contribución y eso, por supuesto, es una meta todavía por obtener. En cuanto a nuestras acciones y el impacto, bueno, en innovación sostenida, la compañía está haciendo inversiones, adquiriendo compañías que hacen productos, ¿cómo se llama? Productos biológicos para reemplazar productos que son solamente químicos. Por eso compramos a Symborg, compramos a Stoller también. También estamos trabajando con otras compañías para producir productos durante épocas que normalmente no se pueden producir, como ha sido con Chevron y con Bunge. En el término de biodiversidad, bueno, algunas de estas compañías han producido una cantidad de agroquímicos muy importante, responsable de la revolución verde, pero ahora sabemos completamente que la sociedad está pidiendo productos que sean amigables con el ambiente, que sean también amigables con las aplicaciones que hacen en el campo. En el área de clima, como dije, estamos interesados en reducir nuestras emisiones. Estamos comenzando por nuestro headquarters en Indianápolis, pero lo vamos a hacer en todos los centros. Y en inclusión, diversidad y equidad estamos aumentando la presencia de mujeres y de minorías, incrementando mujeres en posiciones de decisión y incrementando el interés nuestro por negocios pequeños. Y en esta parte de negocios pequeños, por ejemplo, aquí específicamente en Puerto Rico, la compañía utiliza más del 75% de su presupuesto en compañías puertorriqueñas, no en compañías americanas. Cuando les echo ese cuento a otras personas, todos quedan, ¿what? ¿75%? Y sí, la realidad es que hay una gran cantidad de bienes y servicios que se pueden conseguir en Puerto Rico. Lo que pasa es que entre muchas personas que desarrollan negocios en Puerto Rico existe el prurito de encontrar esos bienes y servicios en los Estados Unidos. Pero si se buscan se encuentran en Puerto Rico. Y si no los hay también la posibilidad de desarrollarlos en Puerto Rico. En términos de inversión, ¿no es cierto? Estamos haciendo una inversión en más o menos del 90% de nuestras soluciones de R&D que sean sostenibles. Estamos haciendo un esfuerzo muy, muy importante para incrementar, lo hemos hecho hasta un 33%, la presencia de las mujeres en las posiciones de dirección. En el centro en Puerto Rico, por ejemplo, hay un centro en Salinas, la persona que es deputy mía es una mujer. Y en el grupo de management, las mujeres representan el 40% de la operación. Hemos sido reconocidos en varios sitios sobre nuestras actividades, tanto en la sociedad como en la producción de nuevos productos que son amigables con el ambiente. Creemos que la innovación sostenible es viable. La vez pasada hice una presentación, pero no sé si estoy hablando a las mismas personas, pero la vez pasada estaba yo hablando que la biotecnología era la tecnología de ayer, ¿no es cierto? Porque hemos hecho muchas promesas en biotecnología, pero no las hemos podido cumplir. Es más, las personas que se oponen a la biotecnología tienen mayor imaginación que lo que realmente ha podido ser la biotecnología, lo cual es lamentable, ¿no es cierto? Porque, o sea, la realidad es que la sociedad está requiriendo muchos más productos de biotecnología que los que la biotecnología ha podido desarrollar. En los retos futuros, estamos trabajando con nuevos productos, nosotros llamamos químicas verdes, que son más amigables con el ambiente. Por supuesto, soluciones innovadoras que garanticen el incremento en los rendimientos. El doctor Negrón hablaba sobre los agricultores y por supuesto la agricultura en Puerto Rico. Y una de las razones por las cuales no hay agricultura en Puerto Rico es porque ser agricultor es difícil, tremendamente difícil, porque no tienen recursos, porque no tienen acceso a la tecnología y porque el trabajo en el campo es duro. Es más fácil llegar a una oficina a las 8 de la mañana y salir a las 4 de la tarde que trabajar desde las 4 de la mañana hasta las 9 de la noche. Entonces, ser agricultores es difícil, pero la tecnología puede ser una herramienta importantísima para que los agricultores puedan tener una mejor manera de vivir. Como les decía, tenemos productos de química verde y ahora hemos desarrollado criterios de sostenibilidad para el desarrollo de nuevos productos. Los nuevos productos deben tener, ¿no es cierto?, bajo peso molecular, no deben ser muy aromáticos, deben ser amigables con las, ¿cómo se llama?, las abejas, y deben tener todos esos características para poder ser aceptables como nuevos productos en el pipeline. En el área de semillas, la compañía tiene dos áreas, una de semillas, una de crop protection. La de semillas, pues obviamente estamos encaminados en la obtención y el enriquecimiento del germoplasma por recombinación genética, porque es la única manera. O sea, por ejemplo, en maíz o en soya, los genes son limitados y la única manera de encontrar nuevas funcionalidades es como en un juego de naipes, tener el mismo juego de naipes y cada vez dar una nueva, ¿cómo se llama? Revolver, ¿no? Todos los naipes y volver a tener una combinación diferente de la organización de los naipes. Y en crop protection, pues se están usando ensayos predictivos. La química moderna no es como la antigua, en que sencillamente se sacaban los productos y solo se veía después cuál era la aplicación. Ahora se puede prever antes de que se saquen los productos cuál va a ser su función. Y entonces aquí tenemos algunos ejemplos. Hemos comparado las metas de desarrollo sostenible de las Naciones Unidas con respecto a las cosas que estamos produciendo. Tenemos pues semillas en arroz, en soya, en sorgo, en canola, en maíz, y con diferentes características relacionadas con la meta número 2, que es cero hambre, el 6 que tiene que ver con el agua limpia y la sanitation, en acción de clima, ¿no es cierto? Y promoviendo la vida en la tierra. En cuanto a productos de crop protection, también hacemos el mismo ejercicio y esos productos tienen esas características con respecto a los goals del desarrollo sostenible de Naciones Unidas. Déjenme presentar aquí algunos productos. Por ejemplo, Reclamel es un nematicida. Los nematicidas, si hubiera aquí agricultores, rápidamente los agricultores me habrían dicho que los nematicidas en general son productos terribles, con un impacto ambiental brutal. Nosotros estamos sacando nematicidas con bajo impacto ambiental. Estamos sacando nuevos herbicidas también con bajo impacto ambiental, como el Greenscore. Fungicidas como Inatrec, que son muy específicos. Incluso productos como Sosdia, que ayudan a mitigar los nuevos estreses a los que están sometidas las plantas, porque con el cambio climático, por supuesto, cada vez hay menos agua, las plantas están sometidas a mayor estrés y este producto ayuda a obtener. O sea, y además estamos produciendo, por ejemplo, semillas como Vorseed, que permiten la aplicación de de herbicidas que tienen gotas más grandes y que tienen menos distribución en la sociedad. Yo creo que esta es básicamente mi presentación. No sé si tienen preguntas. Muy bien.
Well then, thank you, Dr.
Muchas gracias.
Sanchez, for your presentation. Our next speaker is Dr. James Elrich. He will be online with us, so I will begin introducing him. He is the Director for Compassionate Sustainability at the Stanford University School of Medicine, Center for Compassion, Altruism Research and Education under James R. Doughty. Additionally, he is appointed faculty at Singularity University, Senior Fellow at NASA Research Center, and at White House in the Office of Science and Technology Policy, appointed to a joint task force on regenerative infrastructure. Dr. Elrich is a graduate from New York University Gallatin and a medical engineer from Stanford University. He has won several awards for sustainable design and is a researcher and global lecturer on the topic of regenerative neighborhood development. He is co-authored of 2 UN Sustainable Development Global Platform briefs, and with Professor Larry Layfair and Chris Ford from the Center for Design Research at Stanford University. Welcome.
Thank you so much. It's really an honor to be here and to participate. I will go ahead and share my screen. So the topic of my talk today really is about collapsing distances between where high-yield organic food is cultivated and produced to where it is consumed, and specifically related to the idea of how we live and where we live in terms of self-reliant, resilient neighborhood infrastructure. And so I'd like to, again, thank Dr. James Doty at the Center for Compassion, Altruism, Research, and Education at Stanford University, Professor Larry Leifer, and Dr. Chris Ford from the Center for Design Research at Stanford University. My background coming out of New York and after my undergrad at NYU was really looking at the sort of crossroads between technology and nature. And although I had started my career in video game design and development way back when, I was always passionate about food, about healthy eating, about cooking, about community. And I started to look into this whole idea of something called eco-villages. And there's a wonderful definition by Diane and Robert Gilman back from 1991, which essentially says that these are communities that are human scale. They're full-featured settlements where the nature is integrated harmlessly and in such a way that not only does it support human health and development and species development, but also that it can continue on into the indefinite future, meaning this is, for me and for us, the idea of regenerative, the concept that things can continue forward and improve as time goes by. So going back into history, now this is maybe 3,000 years before Mesopotamia, were these incredibly large communities in Eastern Europe, somewhere between Moldova and Ukraine, spanning sometimes over 15,000 residents. And these enclaves, sole purpose really was this idea of soil health. They had animal husbandry, but their focus with the animal husbandry was on returning these nutrients to the soil. They understood, in other words, completely that soil health equated to their health and their ability to cultivate a variety of indigenous cultivars that would allow them, for the most part, to thrive. And yes, once in a while they ate meat for festivals and celebrations, but it wasn't the primary objective of these small plot animal husbandry next to each one of these dwellings. Going along with that logic is the culture around the Caribbean, the Taínos, and the fact that this is a culture that focused on soil health. They focused on cultivation that was in combination with aquaculture and fishing to provide for not only the needs of themselves, but in terms of trading and barter across the islands. And that was a model that existed for a very long time before, of course, Columbus and the European kind of settlers came to turn it into more of a monoculture kind of production. So there's an interesting study that kicked off last year, 2023, from Florida International University, focusing specifically on this idea of the canuco and using basically guava and guano, for fertilizer to really focus on how the soil can be replenished and how the cultivars can feed in a very small plot a family of 4. And now, of course, we look at sort of how we can expand that to neighborhood scale. But these are excellent references for historical references for how to imagine neighborhood scale resiliency in nutrition. So going to the macro scale now, where the world is, we look at these coastal megacities around the world where everything is being sort of imported in, right? And including in Puerto Rico, as mentioned by the previous speakers here, that a good percentage of the food is coming in through ship and an airplane. And so what can we do really to look at this sort of flow, both of food and other kinds of calories, including water and power and data, as well as the export of waste. And all of these start to fall in line, especially going back to the neighborhood scale, with the difference between sort of entropy and syntropy, right? Where we can find opportunities for resiliency. So this is a really telling slide in reference to global food miles and the relation to not just emissions, but our dependency on this global infrastructure for feeding ourselves. The primary issue here is, and we saw it very well during COVID, was not just that people were afraid and started to hoard, not just that individuals and families were doing that, but nation states were also holding back certain amounts of food supplies because they were also concerned about how they were going to feed their populace, right? And also there were some other kinds of things that happened, like there were blockages to the Suez Canal, and even one ship that blocked the Suez Canal during the COVID crisis had caused a cascading effect that shelves and supermarkets around the world started to go empty and bare. And this again creates a negative feedback loop that starts to cause fear. And that's when people start to think differently about a lot of different things, specifically about how they live and where they live. And in urban environments, people started to think, maybe we should start thinking about living outside of cities, suburbs, and peri-urban and even rural areas. So when I'd come to Stanford back in 2012, I was armed with this sort of master's thesis on neighborhood scale regenerative resiliency, and really focusing on this idea of initially the built environment, the concept of the single family passive home, but I really wanted to extend that to this concept of neighborhood scale. And what do we mean by that? That the substrate of a neighborhood scale, typically you have a microgrid, which is a power generating and storage and redistribution model. But I wanted to include in the definition of a microgrid this idea of high yield organic food production coming from diverse, biodiverse, biodynamic, non-till soil methods. Also, the inclusion of water management, which has everything to do with permaculture and planning in that way. And of course, energy generation storage connected to passive homes, smart mobility, and the like. The idea really being neighborhoods that can be islanded themselves, self-reliant, in case of fill-in-the-blank district utility failures. I was deeply inspired back in the early 1990s by the work of Dr. Suzanne Simard on this idea of mycelial mycorrhizal inoculations under the forest floor. And this concept really that these inoculations under the forest floor are a broker. They connect the diverse cultivars across pretty vast distances. Some of these fungal networks can be somewhere between 20 to 30 radial kilometers. So they're giant organisms. They are sentient, they're smart, but they don't have a central brain. Instead, they have intelligence at the point of sensing and are capable of then sharing that information across the wider network. To me, that's a really inspiring model because it is a self-healing network. It sort of competes with itself in a nice way, but at the same time, it's a broker, as I mentioned. It's brokering carbon, nitrogen, phosphorus, sugars, minerals, even water across a network where the cultivars are saying, I have this, I need this. So it's really amazing to imagine under the forest floor what is going on every day around us, and to say, okay, how can we use machine learning and artificial intelligence to begin to explore this concept, right, from a neighborhood design perspective. And really, so how can we look at food production, water management, energy, housing, mobility, all of these under that context, that umbrella of output of one system can become the input of another, right? Very much akin to the mycelial network. Moreover, how can we look at using machine learning to start to understand previously unknown relationships between these siloed infrastructure and component pieces? Again, very much as if we're looking at a network that is continually exploring and learning. That's exactly what a fungal network does. It searches, it finds, it ingests, and it learns, and it continues to expand itself. So in that way, we look at a system that can be self-healing, improving, mitigating against risk, and using it all of these different components at the neighborhood scale to do that. One of the key things about what we're working on is this idea of designing neighborhoods and communities, first and foremost, data-driven about what the land wants, right? It's inverting the model. Traditional real estate, residential real estate planning starts with urban planners and architects with these grand visions. They put out these maps and they plan these great communities in their minds. And then subsequently, somewhere far down the road, come the sort of pesky environmental studies, right? And what really the land is saying that is defying their great sort of edifices of suburban subdivisions. Rather, we think we can use this information cycle in kind of a SimCity virtual manner to sort of create a model that brings nature forward first. So by way of visual example, here is a 60-acre, 25-hectare parcel of fallow farmland. It's zoned by law under agricultural rules, which means you can't touch it for anything other than agricultural purposes currently. But we like to imagine, by visualizing what you could do with that land near suburban or peri-urban or even rural, how you could imagine 400 or 500 families living in social, affordable, dignified housing, pushed to less than 1/3 of that land, but where you can see that more than 2/3 of that land is attributed to biodiverse, biodynamic, non-till soil farming, variety of cultivars, full menu design challenge at the neighborhood scale. You think about that for a minute. As much of the menu that you possibly can derive within the footprint of a residential community, right? So it includes some small animal husbandry, chicken, egg, turkey, whatever the sort of cultural milieu might suggest for that location, in combination also with controlled environment farming and aquaponic and aeroponic systems, as well as water capture, energy generation and storage, waste to resource management. Again, all of those things in symbiosis with humans, with living naturally. Again, when you look at this kind of community, you have to really imagine that when, not if, but when district utilities fail and district services fail for whatever anomaly that occurs, hurricane, earthquake, fire, drought, whatever it may be, that these neighborhoods continue going forward without interruption, right? And in the aggregate can be overproducing these artisanal ingredients. And by artisanal ingredients and by nutrition, what do we mean, really? We're talking about life-affirming ingredients. We're talking about this idea that in the footprint of the neighborhood, you have all of these kinds of elements and components coming together to contribute to soil health, number one, biodiversity, and also this concept that you're defying pests by moving things around in that way. Looking at aquaponic and aeroponic systems, it's an ancient way of fish farming, right? But we use this in a controlled environment that the fish waste right? So freshwater shrimp, crawfish, several different species of freshwater aquaculture, their waste is ammonia. But through a light biological extraction and interaction, it's converted from nitrites to nitrates. And with a low energy water pump, comes and feeds these lattices. And the water then feeds the plants and the cultivars that are dangling with the roots into that nutrient-rich water. The water comes back to the fish tanks fully purified. So you can start to get a picture of what the soil-based, non-till soil farming in the aggregate can produce, the controlled environment year-round, which can be really biodiverse as well. It's not just leafy greens and microgreens. I've seen in my research around the world stunted papaya, amaranth, quinoa, all kinds of very interesting high-protein grains and other kinds of cultivars. And of course, what people can grow next to their doorstep. It's all about food security. And specifically in relation to Puerto Rico and other islands in the Caribbean, this is absolutely the model that we have to be focusing on, as well as other places around the world. In terms of waste digestion, we have been really focused for many years early in our process on black soldier fly larvae, aquatic worms, and other anaerobic digesters that can eat their own weight every day in food and animal waste. But guess what? Those are then fed back to the chickens and the fish and the other kinds of small animals because they are full of omega-3. They're filled with protein and fat, and it's the perfect nutritional input, live input, especially for the chickens and the fish, right? So this concept of designing neighborhoods around year-round, artisanal, delicious ingredients that are bioavailable, okay? Our argument as well is that with those kilometers you saw before of food flying around the world, that the moment your food has to travel, even by truck, from one distance to another, it's beginning to lose, we feel, its nutritional value. Its higher bioavailability is directly where it is cultivated and harvested, prepared, cooked, or preserved, right? But not only that, these communities have a life-affirming, visceral connection to where their food is coming from and that food security. Piazza kitchens, farm-to-table experiences. These are things you don't need language. People can sit at a table, not speak the same language, and be able to enjoy recipes which express history, ancestry. And that connection in designing communities where you're living within nature, not separate from it. And we are very much a part of nature, right? But what was happened over the last, let's say, several hundred years is that we have become further and further disassociated from the natural world and our ecosystems. So this concept of access and agency, right, with having some soil even under your fingernails, is a proven metric for happy longevity, right? So for instance, the blue zone research, which is where people are living to 100, 110 older years around the world. They're living in these small villages, in these communities where they're multi-generational, they're connected to each other, they're connected to their food sources, they have some involvement in the cultivation, in the harvest, in the cooking, in the sharing of these ingredients. And that is an absolute moniker for happy longevity. And that's actually my current affiliation at Stanford, is in the School of Medicine at the Sea Care Institute, which is by how we reduce amygdala response, which is that fight or flight release in your brain that triggers cortisol. And if you can reduce the amygdala response and reduce the cortisol release, you live a longer, healthier, happier life. It's just proven, right? So it's idea of designing the balance of nature as an amenity. It's this idea of education and bringing children up, understanding the full circular story and story arc of life, where people are born, how they're raised, celebrating life moments, understanding the people getting older, then they need sort of elder care, hospice care, and yes, death notices. All of those things happen within a healthy community. You see all of that, and it's not where we're pushing the seniors off to one kind of complex and putting parents with small children into another kind of complex, and again, disassociating ourselves from each other and nature, right? So all of these things are interconnected for happy longevity. One of the important topics I just want to kind of get to here before I finish is this idea of improving and restoring local and regional supply chains, especially around food and nutrition and that bioavailable nutrition, right? So our goal is to look at how we can use these emerging technologies, drone taxis, drone deliveries, autonomous vehicles. Where I am here at Stanford, and all of these technologies are coming, certainly within the next 10 years or sooner. And that means that islands like Puerto Rico are safer because they have an overproduction at the neighborhood scale of their own artisanal ingredients and are able to share and barter and trade within the context of their own island, right? Being able to have that food security is incredibly, incredibly important. So to recap, what we're doing is applying machine learning to this idea of indigenous wisdom. What does the land want and need for species, including us, health, flourishing, biodiversity, bioavailability, and to create resiliency in climate adaptable communities? So I like to say our time is now. This is what we must be doing and focusing on. And I also just want to have a quick shout out to a friend, Adam Greenfader, and this wonderful book, which is Why Puerto Rico Now, right? So there's an important read there on, and some exciting opportunities and news that we'll be sharing shortly. And I look forward to working closely with some of these wonderful, esteemed colleagues on how we can realize residential communities in Puerto Rico that can feed, power, hydrate, digest their own waste, and solve for social affordable insecurity in terms of housing. And so with that, I want to thank you so much for your time, and I look forward to any questions that you might have. Thank you so much.
Dr. Ehrlich, thank you very much. Do we have any questions? Yes, we have one.
Hello, there we go.
Now you go.
Thank you so much. I really appreciated the talk. It's like a dream what you're representing here. My immediate question is, how do you foresee the kind of balance of the internal economics of a village like this? For example, what percentage of the people living in that village would be responsible for the production of food, and how would they be compensated for that responsibility?
Good question. And first and foremost, we see this opportunity to work with local organic farmers and also in education and training young people and older people as well on permaculture and organic and biodynamic farming principles, and to offer a living wage to them. And we do that, and we consider that through this idea of course, mixed socioeconomic level housing access. So you have market rate, you've got middle class, upper middle class, and also affordable and even social access, right? That there's a kind of blend as well to monthly association fees. That those contributions economically into that would pay the living wages for those local farmers to operate and be supportive of that infrastructure. So it's not an obligation or a demand necessarily on the people living in the community, but it's definitely an improvement potentially to people's health by being involved in that way. So that's really the way we would sort of tackle that from an economic perspective. but also we feel that from an educational model and from a health perspective, that there are other benefits that can come from participating. But it's a great question.
Thank you.
Any other questions? That was definitely an important question, because how are we going to manage that? So it's insightful. Thank you very much for your answer, indeed. Thank you, Dr. Erwich, for your presentation. We will now present Dr. Emily Merchant. She is an assistant professor in the Department of Urban Global Public Health at the Rutgers School of Public Health. Dr. Merchant's research focuses on the interface between food environments and human health, using culturally preferred foods as a vehicle for delivering nutrients to underserved populations.
Hi, everyone. Thank you for being here. It is a dream to be here, not just here in beautiful Puerto Rico, but here with all of you having these conversations. We know globally that the human population is rising, that the planet is warming, and that people are hungry. And when we talk about hunger, we not only talk about hunger from the perspective of the quantity of calories that people are consuming, but the quality of calories that people are consuming. Are they culturally relevant calories? Are they nutritious calories? And these problems are made even worse in countries like Puerto Rico and the Caribbean, which have risks of food security in these small island nations. And this was talked about in large extent by other colleagues, so I'm not going to go into this in too much detail, but we know that the high dependence on imported foods, the small economics, the high cost of food, the disproportionate climate risks, and access to land tenure, all made more complicated by the trade and colonial histories of power inequalities in these countries, threaten food insecurity in a way in these island nations that we don't see in the mainland. And I don't need to show this to everyone here. We all know what the hurricanes did in 2017, and these outcomes were seen even more so after the COVID-19 pandemic and other climate situations such as earthquakes and Hurricane Irma. So the promotion of nutrient-rich indigenous and traditional vegetables and medicinal plants can renew the food system and serve both human and ecosystem needs. And here in Puerto Rico, these are foods that were traditionally eaten by the Taino peoples, and they have high value within the local food system. They're nutritionally dense or have medicinal properties, and they have high economic potential, both through sales locally, but also through exportation and potentially through the lower input requirements. Often these crops are habituated to the local ecosystems and require less of those inputs compared to Western introduced crops. They still need things like water and soil and healthy fertilizer, but maybe less so than Western introduced crops. And we have seen the use of a market-first, science-driven model to help bring these culturally relevant foods to the tables of people. And this can be used to build a sustainable and adaptive food system. And so when we think about this market-first approach, science-driven and community resiliency framework, all feeding down into an environmentally and economically sustainable food system. But in order to do this, we need to do both a needs-based and a strength-based assessment of the local food system so that we can bolster both food security and food sovereignty. As many of my colleagues have mentioned already, it's not just bringing food, but it's the empowerment of the people and the appropriateness of the technologies and the foods that are being given to people that is going to be the change agents. And so when you see in this model, we're not only talking about the consumers, but we're also talking about the producers and their right to land tenure, and their right to the access of the inputs that are required. So human dimension research depends on both qualitative and quantitative data collection to perform both these strengths and needs-based assessments. And then this can hinge on existing data, but as was presented by one of our former colleagues, often in spaces like these, in small communities, this existing data doesn't exist. So we need to lean on the collection of new data so that we can understand both what are those strengths within the communities as well as the needs that need to be addressed. As an illustrative example, I'm going to talk about a project that we conducted in Kenya. First, we conducted this strengths and needs-based assessment to understand what were both the barriers and the facilitators to the production and the consumption of African indigenous vegetables in the countries of Kenya and Zambia and Tanzania. And we found that preparation, abundance, affordability, and health were all big drivers for why individuals were consuming these foods. But there were still many barriers that they faced in the consumption and the production, which we were then able to address through various interventions throughout the food system. And so we're going to use this model. This is the Johns Hopkins Food Systems Primer as kind of the model and the illustrative background. And you've heard from many colleagues now today in that box up there in terms of the production, what are different types of innovations that can be done. And these innovations can be done on many scales. It can be biotechnology, but it also can be solar power innovation to run a water pump to bring water to the fields. One of the biggest innovations that these projects did was for seed saving. We talked about seeds and the needs of the imported seeds. In many countries like Kenya too, they also rely on that importation of seeds. Farmers would try to save seeds, but often they would go bad because it was humid and animals would get to them. So one of the biggest innovations that we did was introducing plastic Tupperware containers with the silicone gel desiccant balls to help keep the seeds. This isn't a huge technology. This isn't scary technology. It's just innovative to the problems that we were faced, and it was an appropriate technology for these communities. But we need to think about the rest of the food system as we work on these solutions. Increasing production, or the efficiency of production, the labor for production is important, but so is bringing that food then to the consumer. And often when we think about smallholder farmers, the ability to bring the food to the consumer is quite challenging. How do we get it from our smallholder farm to that market space? And so in this project, we worked a lot on conglomeration, bringing the smallholder farmers together so that we could then pool those resources and get them to a wholesale market. Through this project, we saw the foods going from small sales on the streets to actually being introduced into the grocery stores. And this is a big deal, particularly in Kenya, where the African indigenous vegetables often had a negative social stigma. They were considered to be famine foods. They were considered to be consumed by people of lower resources. But now we're seeing them in grocery stores. Now we're seeing them in high-end restaurants. And so we were able to kind of shift that dynamic of how these foods were perceived within the communities and to really empower and invigorate those local communities. We also then worked on the consumer side. How do we get individuals to eat the foods that are being produced? How do we reshape that social stigma? This is a picture of a solar dryer. So again, low input technology. This is a solar chimney, all using solar energy. And then this is just a drying bed, and you would put screens on top of the vegetables so that you were able to protect it from dust and bugs, and you could see the chickens in the background too. And so you're able to protect it from all of that. And we were in a woman's house in rural Kenya. They had this solar dryer, and so I asked, how are you eating your dried vegetables? Are you enjoying those dried vegetables? How are you using them during the dry season? That's the intention of this, to preserve the vegetables when they're in plenty, to be used in the dry season. And she said, we're not. No one taught us how. And so it stopped here. She had bags of dried vegetables in her house, and she wasn't consuming them. Her family wasn't consuming them. So we created culinary classes and improved recipes with taste and nutrition. And we conducted at the community level cooking classes so that the whole community came out and cooked together, the husbands, the wives, the children. It was very important, and it really highlighted the importance of that human dimension piece, right? We could come in with all of the technologies up in the production space, but if no one then considers the consumers or engages the consumers in the conversations, there could be a missing link in that dialogue. And we saw that this program had a protective effect during that dry season, as evidenced by shifts in consumer behavior. So pre-intervention, we saw most of the individuals getting their food from the village market, so their hyper-local food system. And then after the intervention, and I should say that this was conducted during the rainy season, so during the harvest season, and this was conducted during the dry season, so the hunger season. We saw a shift from that village market to that town market. And we saw more for the individuals we saw that shift more for the individuals who didn't receive the intervention. We also saw a protective shift in diet quality, and so as measured by dietary diversity score. So the individuals who received both the nutrition and the production intervention had an improved dietary diversity score relative to the other groups. And we also saw this in terms of project sustainability. And so it's important, we've talked about the economics in several of these different talks. How do these projects survive past the funding season of the grant or without grant money? And we were able to see that here with our urban agricultural project, that it is still flourishing and thriving and even growing to include the youth. When it first started, it was just a women's project, and now it's also a youth project as well. And so we were able to see sustainability of the program goals past the intervention period. So what is next? Well, now we are here all together talking about how we can bring this community-based participatory program to farming households to improve food security, access, and availability in Puerto Rico and in the Caribbean. And we're going to do this by leveraging existing frameworks as one way. We need to leverage again, thinking about the strengths and needs-based assessment, we need to leverage the strengths that already exist within Puerto Rico and the Caribbean. So these are just a few examples of organizations that are here in Puerto Rico. And this is a model that was developed by farmers in Puerto Rico about what they imagine a climate-resilient food system to look like. So in conclusion, the domestic farmers play a key role in producing food in Puerto Rico, and we're going to need to use a social ecological lens to conduct a strength and needs-based assessment, and we will do this by leveraging pre-existing frameworks to build a sustainable and adaptive food system. I have many acknowledgments, of course, but mostly thank you for listening.
For Dr. Merchant, we have a question here.
Thank you for your excellent presentation and for bringing the importance of added value to, if we're looking for food security, we need to develop more spaces so we can hold those products more time and added the value. Even in Puerto Rico, we are looking for lyophilization process, and that needs cold. So we are in a tropic, so we need to see solar as the option. And that is what the whole world is moving to. Even I've been in Taiwan, and they are doing the same thing, larger scales. And it's very similar. Thank you for bringing that to this point of the conversation. My question is about what do we need, what do you think we need in the Caribbean or in Puerto Rico per se? And thank you for being so updated to our information and our process of life here. And what do you think we need as to build a framework in Puerto Rico? So we need funds to do that, but where they can come. We have several programs, a lot of programs. Sometimes we gave back to the United States a lot of money that we haven't been able to use. And how can you think about the framework that we can deter those funds, even if we are not willing to use it, or for X or Y reasons, to improve this kind of technology, if you want to say it, or way of living?
The first thing that comes to my mind, a sense of a consortium of all of the major actors that are doing work, not only in Puerto Rico, but in the Caribbean as a whole, so that we can all be working together as a large. So that's one thing that I think of, is how do we get all of the different organizations in the same space having conversations. Am I off? I am off, sorry. To reduce redundancy. Redundancy is great. That helps build resiliency. But reduce redundancy from the perspective that the redundancy is thoughtful. It's purposeful to build that redundancy, not redundant in a way that because we don't know what everyone else is doing. So I think of a consortium. I also think of data collection. I feel like whenever someone's like, what should we do? I'm like, I don't know. We need to go collect data so we can find out. So I think of data collection, which is expensive. So if you're looking to spend money, because I think that's part of what I'm hearing, that sometimes there is work that happens, but there's extra funds that then get sent back from the grant. And so thinking of ways to collect data to help identify those things that we could be doing and those ways in which we could be growing. Thank you for the great question.
Any other questions? Thank you very much, Dr. Merchant. Well, it's time for a coffee break. We will have 10 minutes. We invite you to enjoy the refreshments outside. We will be back at 3:40. Yes? Okay, see you later.
su segunda llamada. Segunda llamada. This is the second call. The second call. Esta es su tercera y última llamada. Tercera y última llamada. Favor de poner sus dispositivos electrónicos en silencio. This is the third and last call. The third and last call. Please put your electronic devices in silence. Thank you.
Greetings, welcome back. We will now continue the presentations with Dr. Diane Beckles. She is a professor in the Department of Plant Science at the University of California, Davis. She is an integrative post-harvest biologist interested in discovering genes that influence the quality of fruit, vegetables, and cereals. Dr. Beckles received her BS, bachelor's degree, in biotechnology from the University of Sheffield in the UK, and her PhD from the University of Cambridge, also in UK, followed by a postdoctoral training at DuPont Agricultural Biotechnology in the United States. She is the author of over 80 research articles, has given 41 national and 33 international talks. Dr. Beckles was awarded an U.S.-Israeli faculty fellowship, and for her demonstrated leadership in student mentoring and advising, received a UC Davis Graduate Program Mentoring Award, and was selected as a Chancellor's Fellow for Diversity, Equity, and Inclusion, a UC Davis Advanced Faculty Scholar, a UC Davis Accelerated Teaching Fellow, and an Association for American Colleges and Universities STEM Innovation Fellow. She was also featured in a Cell Mentor, 1,000 Inspiring Black Scientists in America. Dr. Beckles is happiest swimming with the green turtles that live in Carlisle Bay, Barbados, the country she was born.
Thank you. Buenos días. Lo siento, but no habla español. I want to talk about biotechnology, and I want to talk about it in the context of food security in the Caribbean. I do not believe that biotechnology will solve all our problems, but I am concerned that some people think it will be a problem and not potentially a solution. So my talk is aimed at the younger people here who may not be experts in biotech, and I hope that I can give you reason to think that this technology can potentially have use in the Caribbean. So we'll talk really quickly about some of the challenges faced in agriculture, biotechnology, what it is, some genomic modifications, and then examples that could be practical and useful for the Caribbean. And then, as always, we have to look at the strengths and weaknesses of any technology. So climate change is real, and just the unpredictability of weather makes it very challenging for farmers. Populations are growing and growing rapidly, and some people even think we may get to a point where we may be unable to match population growth with our capacity to produce food. And the food system's becoming really complicated because there's not just population growth, but as humans, we have really intersecting, sometimes opposing requirements of our food. But about 10 years ago, people came up with a rough number and said, by the year 2050, we have to double food production, and that will require the use of technology. So for you, we want to make sure that we can save native plants so that you can have foods that are culturally relevant for you, and which are, not only culturally relevant, but sustaining and nourishing. So the National Academies of Sciences put out a white paper and described the technologies that they think would be important for food security in the future. And they said that, there's a wide range of solutions are going to be needed, but advances in genetic evaluation and modifying plants, including by gene editing, must be part of that conversation. And so that's where I stand. So biotechnology is basically about using what we know about genes and the traits that they encode for the purpose of improving plants, and more specifically crops. And so one of the questions I get asked a lot is, do we really need to genetically modify plants when nature has already created this beautiful diversity? And you've seen pictures like this already today. and Dr. Sonia Peter, who was my chemistry teacher in high school, by the way. Treat your teachers well because you never know if you'll see them in later life. There's this beautiful biodiversity that is just, I mean, look at it. It's wonderful. And we want to preserve it, and we want to use it. But we also would like to have other solutions. So what has happened over millennia as we move towards developing our modern crops? We've thrown away a lot of those genes that make up the diversity that we see, right? So that the modern tomato, which I'll use as an example, and Dr. Giovannoni, like it was just wonderful seeing all that tomato work today. What we have now is a very genetically narrow cultivar. And these cultivars that we grow, a lot of them are very susceptible to, adverse environments. And so it would be useful if we could go back to some of the diversity that exists in nature and pull those genes in, as we saw so beautifully done today by Dr. Jim. But we're gonna have to do it really quickly, and we have to do it efficiently. Because of all the challenges, our improved crops need to be resilient to stress. We can't afford to use all the inputs that we currently use. It's expensive, in some cases environmentally very damaging. And we want these new crops to be optimal in terms of their nutrition. And we want the quality to be excellent. And for fruit and vegetables, it's really important that the shelf life be as long as possible to match the supply chain. So what this will require is like a fundamental understanding of the genes, right? DNA encodes as the blueprint for the traits, so the things that we see, the form, function, the taste, the size, to connect between genes and morphology and traits. So the phenotype, which describes different aspects of traits, is determined by the environment, but also by the genotype. And when we talk about genotype, we're really talking about DNA. So these two elements that we're discussing. So for the environment, we just said that we have oftentimes not a lot of control over that. And it's becoming increasingly more hostile towards the way that we grow plants. What we can have some control over is the genotype, and that's where I'm going to focus my attention. So we can, within the context of what environment that we're looking at, we can try to control elements of it so that we can maximize the yield of plants. But again, farmers are always looking out for better cultivars with a higher yield. That will behave in the way they need them to within the environment that they're growing those plants in. So a little bit of genetic lessons. So we said that DNA encodes the blueprint of life. It determines the traits. But the information in that DNA has to go through a couple steps to make proteins. And we consider proteins to be the workhorses of the cells. They are the ones that actually make a lot of the stuff. And so there's this information transfer. But all of these steps are regulated. They may be sped up or slowed down. Some of these intermediates may be degraded. And each of these steps, in turn, are determined by the products of other genes. And so I'm going to go back to this picture here. A lot of the differences that we see in the form and the colors are decided by little changes in DNA that are determining these particular traits. So little changes in all of the genes that perhaps may be part of a pathway or other genes that may regulate them. So again, you will say, see, well, I said, Diane, there's enough genetic diversity that exists. We don't need to do any more genetic modification. I can go to the supermarket and buy stuff, and I can get heirloom varieties. Well, let's just first say that the stuff we buy in the supermarkets is already genetically modified. These are not natural. They will not survive in nature. They were produced by humans. So these are our natural fruits and vegetables. So on the top corner, we have the bananas. Then we have like the undomesticated eggplants, tomatoes, apple, kiwi, avocado. This isn't a carrot, like an original wild carrot. Right? It's not so nice looking, is it? Okay, yeah. And tiny little strawberries. So what happened was that you had these little mistakes or changes in the DNA that existed in these organisms. And that led to differences in traits and to all that diversity. So if, as Dr. Giovannoni was saying, you were able to sequence all of different relatives, you'll be able to find out where you had like changes in DNA sequence. So this is an example. So at the top line, you see where it says cat, cat, cat. That's the DNA sequence, and it encodes protein, which is shown in blue. And if you just get little changes in those letters, you can have very different proteins produced, and those proteins can then lead to differences in the form and function of genes. And so scientists have been really good at making connections between genes and some of the traits that we see that differ between wild and modern cultivars. So for example, people have found genes that are responsible for fruit size or fruit shape. But then you'll say to me, why do we have to know these genes? Like, forget about these tomatoes and maize, and they make up, I think 6 species make up about 50 to 60% of all the calories that we eat. And as Dr. Peter was saying, there are thousands of species that are edible that we completely ignore, and those species are already adapted to poor environments. So why don't we go there? And I say, yes, we can do that, we should do that, but we still also need to make a lot of what we eat more robust. There'll never be a situation where we're not going to be eating potatoes or maize or tomatoes. So we still need continuous crop improvement. And one of the ways we can do this is by gene editing. It's a very nice, exciting way to make changes in DNA because it's so specific. So what you need is a DNA scissors, it's called Cas9, and a little piece of DNA sequence that matches a gene that you want to modify. So you introduce those into a cell. The DNA scissors will match that little strand to a part of the genome that it recognizes, and it will make a cut. But the cell will repair that cut, and then it may make a mistake, and then you have a mutation or a difference in DNA sequence, which can lead to a new protein and a new trait. So how do we get the little DNA scissors and guide into the plant cell? We use a bacteria called Agrobacterium, which has evolved a natural way of modifying plants. So we put our Cas9 and guide into a little plasmid, put it into the bacteria, and it does the work for us. It goes in and it inserts that into the genome. And you may think, well, that's weird. No, it's not. This evolved over millions of years to the point where, like, all sweet potato cultivars have pieces of Agrobacterium DNA embedded in the genome because this process of lateral transfer has been happening naturally over the years. So you say, well, Diane, selective breeding, conventional breeding has worked really well for hundreds of years. Let's just keep doing it. And I say, yes, we should do that. There are lots of traits that work well with conventional breeding. But you know what? There's some traits in some organisms where we can get a new feature or get crop improvement a lot faster with gene editing. So we can use them side by side. And so the gene editing is something that a lot of people are excited about, and the primary reason is because it's easy and because it's really cheap and fast. And because it's cheap, it means that labs that don't have a lot of money or countries that don't have a lot of money can have access to this technology. And that's the thing that excites me most. I was inspired to become a plant scientist because I wanted to do biotechnology, 30, 35 years ago. And now it's come into reality. So lots of advantages to this technology. And if you wanted to start a really small scale project, just with undergraduate students and maybe a master's student, this is about the amount of money that you would need, and this would be the timeline before you could get like a new trait, maybe even less than that. So some very exciting things have been done because the gene editing is such a precise way of introducing changes. It's possible to generate lines that vary incrementally in a particular trait. So here's an example where people were able to have fruit that just varied in size, and that can be very useful. And then what was really cool was that people actually did sort of a reverse engineering. So they took a wild species of tomato and they said, you know what, we've identified a lot of genes that we know differ between wild species and domesticated species. Let's go back and change these. And they did that for about 5 or 6. And what they found was these really beautiful synergistic reactions that meant that you got traits that you couldn't predict. So higher yield because the fruit were larger, there were more produced, and the lycopene content was higher. So there's even value to like making multiple changes in genes in the same background. People have decided to create tomato fruit that may be more amenable to indoor agriculture or vertical agriculture. And what's really cool too is, again, the idea of going to wild undomesticated species and modifying genes that we know are important for ag. So this brown cherry, there are efforts on the foot to sort of make this amenable to agriculture, so it'd be less wild and weedy. So who's adopting this? Companies, because it sounds really fancy and different. China's put a lot of money into gene editing because they want to feed its population, and they see that as a very pressing problem. But what's really cool is in Africa, there's like this beautiful work happening at the IITA Center, where they're focusing on staples like banana and yucca and yam and sweet potato, like crops that are important to the Caribbean as well. So we don't even have to do the groundwork or the beginning work. It's being done elsewhere. Anthracnose has always been a problem in yam since I was a kid. At the IITA, they're working on that, and yam mosaic virus. And yam is a very important food security crop, but it just deteriorates really quickly post-harvest. People are working on making cassava cyanide-free. As a matter of fact, they were able to do it, increasing the nutritional value of sweet potatoes. We love it, we're gonna eat it, might not make it better. Increasing virus resistance in bananas in Cavendish. And recently there's a company in England that are set to sell these bananas that may not brown so easily. Think about that. I hate brown bananas. Just to not see that, this is gonna be fantastic. It will reduce food loss and food waste. And there's a group in Africa also work to reduce post-harvest deterioration in cassava, which is a serious problem. Happens between 1 and 3 days after harvest, which is a real issue because cassava is so important for food security in so many countries and so many regions. And also using gene editing to make cassava flour, really important if we are gonna develop GMO-free crops. So I probably should explain that, but I don't have the time. But this is an exciting development. And a lot of it is happening in Africa. So you ask the question again, can this technology be relevant to the Caribbean? I say yes. If they can do this in Africa, we can learn from what they're doing. We don't even have to repeat the work. We could probably just ask for tissue culture products of the technology that they're working on. So I do think this is important, and you've seen this before from Dr. Juan. We're so dependent on importing food in the Caribbean. But now there are efforts on the way, as Dr. Peter talked about, to reduce reliance and dependency on imported foods. But there are problems associated with growing cassavas and yams. They have disease issues. And if we could have new varieties produced that will have less issues with diseases, then that could make our backyard agriculture more profitable or feasible. What biotechnology cannot do is solve the issue of politics, which is huge. And I read this essay, and it was such a punch to the gut. And so I can't talk about this, but the fact that politicians have such a huge say in what happens on this island as far as food security goes is tough to swallow. But still, it means that you have to invest in agriculture here, even if the control is elsewhere. So I think I have a couple minutes to just talk really quickly about work that we do in my lab. So one of the projects that we've been working on is with potato. So a traditional potato breeding program takes about 16 to 17 years. So we had this idea of, because Dr. Giovannoni said, to get people interested, you have to show them something that's going to excite or, get them to see the technology as being relevant and useful. So what we're working on and have produced, although it's not commercially viable just yet, are these potato lines that are effectively higher in fiber. They have starch that's difficult to break down. And potatoes usually are rapidly broken down to sugars. The starch is rapidly broken down to sugars, both in our gut, so it's a problem if you're diabetic, but also when you try to store after harvest in the cold. It breaks down and it causes sugar production, and when you fry those potatoes, you can get the formation of acrylamide. So we had this project just led by a master's student and with an army of undergrads and very little funding, and we asked, can we produce these potatoes and show the world that you really don't need a huge amount of money and that you don't need a lot of technology, and we could just do this very cheaply, and we did, and people were interested and excited about it. So hopefully we've got a little bit of money to try and take the project along, and hopefully we can bring the public in and engage them with that. Some work that we're doing is to look at fruit ripening. As Dr. Giovannoni said, it's really important for reducing food loss and waste. If you can control the rate at which fruit ripens, it gives you a bigger window for marketing, for shelf life. Okay, the flavor may suck, but at least it won't go, it wouldn't be rotten and you have to throw it away. So one of my students, she's looking at making edits in the promoter region of the gene. So that's the part, you can think of it as a volume button, right? Depending on what sequence is there, the gene may be produced at high levels or low levels. And so she made lots and lots of edits in that area. The idea would be that you can produce lines that vary incrementally in the time of ripening. So, at one point you may want a fruit to ripen quickly, another time you want to make it ripen slowly, and sometimes you may get some interactions that you may not be able to predict. So we're very, very early in the project, but we do have lines that vary. Some are higher and lower in expression, and they do have differences in how firm they are, the color, and the amount of sugars, and that's Jackie right there. So I'll just sum up by saying that, one of the reasons people are so excited by CRISPR is because, it's cheap, it's fast, it allows for people who don't traditionally have access to the big technologies to be able to do just that. But, we do have to be cautious. Sometimes we see traits in the greenhouse, and when we put those plants in the field, the traits may disappear. There are lots of genes that control important agronomic traits that we've not identified. So, in that case, targeted CRISPR would not work so well. And input traits like stress resistance, again, not that many targets, but with a lot of basic research that's growing. The patent landscape is murky, and we still don't know how the public will react to these projects, to these plants. And the European Union still regulates gene-edited crops as GMOs. But what's really exciting is that crops that are really hard to breed, things like sweet potato and yam, we can now make root changes at a really fast rate. and we can think about the kind of things that consumers want. And so developing countries, we're in the game. We can take advantage of this technology. And then for basic science, we can also unravel some important basic biology using this technology. And I'm really hoping that the public backlash will not be as ferocious as when we were producing herbicide-resistant crops. And the last thing I'll say is that whenever we make genetic or agronomic changes for improving crops or agriculture, it is critically important that we take the cultural component into account. And you've heard this many times. I'm a biotechnologist. I live in genes. I'm happy with gene sequence, but I know this for sure. And so while I don't have the expertise to do this, I really respect my colleagues who are working on that front. This is my lab, and thank you for your attention.
Thank you, Dr. Beckles. Do we have questions? Yes, we have.
I think you should have expected this. I have to admit that I am a novice when it comes to the CRISPR technology. But I happen to believe, as a scientist, that mathematical probability allows us to predict potential outcomes. Yes, the bell-shaped distribution. So I'm wondering if the CRISPR technology has advanced to the point to where the randomness of that potential outcome is no longer an issue, or is it that you are embracing that randomness?
I'm going to answer your question by not answering your question. Because the one thing that plant breeders and people who value biodiversity, you love that randomness. You love that unpredictability. You say this is nature, right? And I say that's the same thing that's happening here. We just bring two different elements of nature together, and we revel in that unpredictability. But there are these now these programmable Cas9s that are available that may help to reduce that. So that's how I will answer your question. Did I say what you said?
I think we don't have any more questions.
Okay.
Thank you, Dr. Beckles,
Thank you.
for your presentation. We will now invite Dr. Loren Rodriguez Rivera. Her expertise is focused on zoonotic diseases caused by viruses and bacteria. She earned her bachelor's in microbiology from the Universidad of Puerto Rico Humacao campus, completed a PhD in food science with minors in infectious diseases and microbiology from Cornell University. Her postdoctoral research at Texas A&M University focused on antimicrobial susceptibility of foodborne pathogens in wild and domestic animals. Since then, she had the opportunity to work at the Centers of Disease and Control and Prevention and the Department of State and Human Services in Texas. Currently, she is a senior research scientist studying the SARS-CoV-2 variant diversity in Puerto Rico. Welcome.
Thank you. Thank you for the introduction. Can you hear me very well? Yeah? Good. So today I'm going to be talking about food safety as a cornerstone for sustainable food security. A little bit about myself. These are my grad student years. I was very happy when I was a grad student. I know that some grad students are not as lucky, right? But I was very happy when I was a grad student. So on the left side, you can see me in a dairy farm. It was in the middle of winter in Ithaca. So you can see a cow pie in there. I collected samples from cattle feces, And after that, I brought them to the lab, which is the picture on the right side, where we detected Salmonella, E. coli, Listeria, and other foodborne pathogens. So to me, and hopefully by the end of this talk, you will have a little bit of enthusiasm about food safety. To me, food safety is everything, right? So food security is achieved when foods are safe, nutritious, accessible, and available. Food security resides in the middle of those. I know that there are more factors that impact food security, but hopefully today you will appreciate better what is the link between food safety and food security. So it has been estimated that annually there are 600 million cases of foodborne illnesses worldwide. These foodborne illnesses are caused by a variety of foodborne pathogens. Also, there are 420,000 deaths in the world caused by foodborne illnesses. It is very important to highlight that 1/3 of these deaths are children under 5. So you could imagine that in many developing countries, those populations of little kids are basically ended by foodborne illnesses. There also have been estimates on the economic losses every year due to foodborne illnesses. So it has been estimated that there are $100 billion lost each year. And these costs are costs in productivity, people missing work, also costs in treatment of those foodborne illnesses. And the culprits of these illnesses worldwide are norovirus and Campylobacter. Those are the most cases worldwide. On the other hand, we have estimates in the US. So 9.4 million cases in the US are estimated to be caused by 31 foodborne pathogens. That includes viruses, bacteria, and parasites. But if you account for the diseases that are caused by unknown or unspecified pathogens, agents, this number could go up to 48 million cases. Likewise, you have 1,300 deaths every year due to those 31 pathogens. But if you account the unspecified agents, it could go up to 3,000. So in the US, there are losses, economic losses too, that account for $17.6 billion a year. So the most common pathogens that cause all these diseases and deaths are norovirus, Salmonella, Clostridium, and Campylobacter. Norovirus causes around 5 million cases, as opposed as Salmonella, Clostridium, and Campylobacter, which are the closest to that one, and each one are nearly estimated to cause 1 million cases. So I wanted to take this opportunity to point out that these, of course, are estimates. If you account for reported cases, there are 40,000, for example, Salmonella or Salmonellosis cases a year. So you could see the gap between the estimation of 1 million cases versus the actual reported cases. And it's because Salmonella tends to be a self-limiting disease, meaning that most people just have Salmonella, they have a little bit of stomach ache, probably. Sometimes they don't even have like super bad symptoms, so they don't even go to the doctor. So that's why it's severely underreported. And that happens to other pathogens too. So of course we're in Puerto Rico, right? So I have to show some numbers foodborne and waterborne illnesses in Puerto Rico. And pardon me, I don't have the numbers of just foodborne illnesses. So this is the report from the Department of Health. They put together this report every week, and they report the waterborne as well as the foodborne diseases. So this is a report for the epidemiologic week 52 in 2023, and it was published on January 4th, 2024. And as you can see, on the x-axis is the age of the people that had gotten sick because of foodborne and waterborne illnesses. As you can see, very similarly to the data reported worldwide, children under 4 are the ones that get the most disease here in Puerto Rico. And I want to point out the agents that are reported in this document. So the most cases in Puerto Rico are caused by Salmonella, and the second most common agent that causes waterborne and foodborne diseases is unknown. So researchers, there's an opportunity there. So the third most common agent is norovirus. So I wanted to highlight here that we have a variety of pathogens reported in Puerto Rico. And they're reported to the Centers for Disease Control through different surveillance systems. So the first surveillance system that is making this possible is the National Outbreak Reporting System, or NORS, which focuses mostly in foodborne pathogens. But you can also have the NNDSS, which is the National Notifiable Disease Surveillance System, which is a passive surveillance. So if you get sick with a specific food, you will go to your doctor, and then your doctor will report that you are sick to the Department of Health and also the CDC. And interestingly, PulseNet is another surveillance system that the CDC had, and what they do is that they looked at these pathogens in sort of like a different way. They try to match pathogens from the environment versus animals versus foods through whole genome sequencing and other molecular techniques. So there are other programs within PulseNet that had the opportunity for researchers to kind of like join and send samples to them. So these are the 3 surveillance programs that basically feed the reports that are being put together in Puerto Rico. There's an extra surveillance system that the CDC had, which is the active bacterial core surveillance, which is where they actively go into communities and call people and ask about, have you had stomachache, diarrhea, and all these symptoms, so they can, track foodborne illnesses and other illnesses too, even though people had not seek medical attention. But that's only in like 10 parts of the US. So I wanted to highlight here that all these bugs and pathogens that I've described are biological hazards, right? I explained a little bit about the unknown or unspecified agents that cause foodborne illnesses. This could be chemical, this could be physical hazards, but they could be also biological hazards that are unknown, still unknown. But in this talk, I just want to focus on the biological hazards. So the dissemination of these biological hazards make our food unsafe, and this could happen throughout the food system from the farm to fork, because if you think about it, some irrigation systems in the different farms could carry on these pathogens from one place to another, and they can end up in produce and other different foods that we consume. This could also happen at the level of transportation, storing, food production, right? And it could also happen at the consumer level, specifically on cross-contamination. So these unsafe foods could cause foodborne diseases. And in developing countries, these foodborne diseases could cause malabsorption of nutrients because, as you know, or as you may know, these foodborne pathogens could have sequelae and they could have long-term effects on people. And this malnutrition, this malabsorption of nutrients could cause malnutrition, stunting in kids, increased comorbidity, and so on and so forth. So unsafe food creates a cycle of disease and malnutrition, particularly affecting infants, as I mentioned before, young children, the elderly, and the immunocompromised. So why I chose to talk about food safety is because foodborne illnesses contribute to malnutrition, as I mentioned before, but also because foodborne illnesses can have a lifelong negative health effects on people. For instance, salmonella could have an acute effect on people, right? When you consume food that is contaminated with salmonella, you can get diarrhea, you can get fever, but if it turns out to be invasive, it could cause septicemia. And on the long run, you can get arthritis and other conditions just because you got salmonella. And recently, there have been some studies of these foodborne pathogens where it has been discovered that some of them produce some toxins that interfere with people's cells cycle. So if you think about it, cell cycles are linked to cancer, right? So you can do the math. I'm not speculating here, I'm just saying that it has been discovered that they had an effect on cell cycle. Also, another example is Campylobacter, which could cause Guillain-Barré to the people that had gotten it before. But more importantly, foodborne illnesses are preventable. As I mentioned before, foodborne pathogens can be going from the farm to the table, but at any point in that process, there are measures that could be taken in order to prevent foodborne illnesses from happening. And they are also an economic burden. So in terms of food safety, if we implement food safety in a good way, we can remove barriers to economic growth and has a global impact. Maybe I am very optimistic. I like to think about myself as not a naive person, but I think that food safety is very important in achieving food security. So the FAO estimated that around 1/3 of the food produced for human consumption in the world every year is lost or wasted. So if you think about this, there's not only foodborne pathogens, there's also spoilage pathogens, right? Spoilage organisms that could cause food to go bad. So in the aforementioned FAO estimates on waste, they do not include unsafe food that is being consumed. So to quote Crean and Ayalou, they said that we are probably significantly overestimating the global food supply because the unsafe food is not extracted from the data. And I just wanted to point this out because every time that there's an outbreak, a foodborne outbreak, there's also recalls if a food can be pointed as the source or culprit of this foodborne outbreak. So those recalls cost money, and also that food never gets to the consumer. So that's why they're said that there is an overestimation of global food supply. Because if you think about it, by 2050, as the other speakers had mentioned before, we had to feed 9 billion people, a little bit over 9 billion people, right? And we had to do it in a fast manner and we have to do it in a safe manner too. So the last point that I wanted to make with my talk is natural disasters. We had been listening about different natural disasters in Puerto Rico. So perhaps hurricane and earthquakes exacerbates the problems that we had with foodborne illnesses, because as you can see, the report that I showed before was in a year that we didn't have major hurricanes or major earthquakes, right? And there were about 800 cases. But by the time we had Maria, we had a surge in all these diseases, including leptospirosis, and is a bit concerning. Also because there are power surges, and if you don't have power, of course, you don't have refrigeration, you have a lot of problems with distribution and also storage of these foods. So these natural disasters highlights that there's going to be a spoilage and perhaps unsafe foods that could probably reach the consumers. So if anybody had a more recent estimate on this, I really appreciate it. But I read the Department of Agriculture in Puerto Rico estimated that losses in this sector after Hurricane Maria were about $2,000 million. And if you think about this, this was in the agricultural sector. It was because there were so many products that were lost. Even I'm thinking about, I was talking to a colleague not long ago that had to do some investigations, some research during these outbreaks after Maria, and it was challenging because people got food from different programs, but they didn't have a way to store them, right? So little interventions, just as this one that I'm showing, this is just in the webpage, in the Department of Health webpage. This is just a little intervention where they just showed what people should do if they lose energy during a hurricane or after a hurricane or any other natural disaster. So what I wanted to highlight is that sometimes we think about ourselves as, I need to come up with a big solution. So sometimes tiny solutions or interventions like this could help a lot of people during these times. And lastly, I just wanted to say that food safety and food security are public health concerns. My field is in, my favorite field, right, is in food safety, but I also worked in public health, so I know how those two things are really connected. And in terms of food security, they're interrelated too. So that also highlights that we need to take an approach like the One Health approach, right? Where we can work with the environment, work with the animals, with people, in order to keep people safe and healthy. So in conclusion, as you know, there are new pathogens and hazards that are regularly emerging. One example is SARS-CoV-2 that caused COVID-19, which is not a foodborne pathogen, right? But it's an emerging disease. So the same happened with regular, everyday Salmonella, E. coli. They are evolving, and they could acquire genes that could make them more antibiotic resistant, and they could make us sick. So effective solutions to improve food safety require a multidisciplinary and multi-stakeholder approach. And hopefully we can achieve this through these 4 areas. We can increase awareness of food safety issues and impacts and measures to reduce food safety risks. We can build local research capacity and conduct research on regional food safety challenges. We can also help to develop policies that enable conditions for food safety research, translation, and practice. And finally, we can do some training as scientists, we can accelerate translational research technologies and practices. So I would like to thank Alok and Dr. Haley Oliver and Dr. Whitman. They're always very kind with me whenever I have questions regarding various topics, and the Science, Technology, and Research Trust, which is where I work. Thank you. Are there any questions?
Hi, thank you so much for your presentation. I wanted to ask you, based on your recommendations and your conclusions, I know that a big thing that's coming up is into this year with the FSMA, the Food Safety Modernization Act, and how that's going to impact farmers on the island. And so I know it impacts the entire supply chain, but it seems like the concern is that how those regulations might make it harder for farmers to become certified, or we have a big informal economy here. So I'm wondering, how would you think about like the upcoming FSMA and how to work in a multidisciplinary way, so that way it doesn't become even more of an economic challenge or burden onto the producers? That's
a very good question. So basically, FSMA had been there for a long time, since I almost graduated and went into my postdoc since 2013, about 10 years, right? So they were working on that transition that you're talking about, so people can get informed about the regulations and everything. So there's a lot of groups that had collaborations, universities with actual farmers and food production settings, and I can give you, some contacts. One of my dear friends, his job is basically that. He works in an agricultural land-grant university, and he developed these trainings for farmers, for people, so the transition could be better for them, so they could understand and embrace FSMA as it is. Thank you.
Thank you.
There's another question.
Hi, thank you. You really convinced me that is important.
Awesome.
Right? I appreciate your presentation.
Thank you.
How would you, with all your experience, go about creating a robust food safety program for your organization with the challenges that a country like Puerto Rico faces culturally? And there's a lot of the scientists here that spoke about the limited data that is available here. Mm-hmm. And in addition to that, you've had so many challenges such as Hurricane Maria, and that's so fresh in people's mind. How would you leverage some of these things that you know about this country?
So part of my answer will be like the professor here. She said that when there's no data and you don't know what to do, you have to collect data, right? And you have to spend money for doing that, so what I would think is that there are so many gaps right now in terms of food safety in Puerto Rico that could be filled with collecting data, not only questionnaires, but doing small kind of like projects where we can collect data regarding farms, different environments, and in order to do that, I know that funding is required. So there are several ways people can do that, and perhaps my workplace had some opportunities for scientists to do several things, to get some seed money to do research, right? So that's a way to start. Another thing is to write actual, proposals for USDA, for the Centers for Produce Safety, which gives money to scientists that would like to generate data that doesn't really exist. So I would say that collaboration and try to get a little bit of funding to start, like a jump start for those. That's what I think could be done.
Could I ask a follow-up question?
Yeah.
How do you convince then those the farmers that have less funding to then go and do that? Or, and I'm new here in Puerto Rico, so I don't know what the collaboration space looks like as well. So if you could speak a little bit to that, I would be interested.
Yeah, so I think I'm talking about my experience, right? I had in the past when I was a grad student to kind of like collect samples from different farms, right? So what we basically did was, it was not a burden to the farmer per se. We didn't ask them to invest any money. We were just there with the veterinarians and collect our samples and, you know, bring them to the lab and kind of like try to show them the long-term benefit of that. There could be resistance, right? Because not many farmers would think that, you know, looking for salmonella in their cow's poop will be a good idea, right? But I think, I don't think they had to invest a lot in order to get what our data would provide to them. I don't know if that makes sense, if I answer your question.
Yes, then who invests then?
So it has to be funding from the researcher, right? That's what I was trying to say. Sorry, it wasn't clear. Yes, so it doesn't come. The money doesn't come from the farmer. It comes from the actual researcher or people that wanted to generate that data.
Okay. Thank you.
You're welcome.
I'm curious about a slide that you present about foodborne illness and the amount of cases of salmonella, and you talk about 1 to 4 years the children that were infected. Those products or food that cause the illness. Did you know which kind of food it was? It was fresh food. It was processed food, milk.
Yeah, so what I can tell you, the report is very long. So some of those outbreaks, for instance, are caused by meat.
Meat?
Yes, some of them are caused by other sources. I'm trying to remember, because you know, milk industry here has monitoring systems, right? But I would say that, you know, I was talking to some colleagues, and you know, some of the places where you can actually get foodborne illnesses from or products is meat. But for sure, I know that some of these outbreaks are caused by meat. I don't know if it was locally produced meat or if it was imported meat. But what I would say is that meat will normally have E. coli and salmonella and all those bugs. But I can find more answers for you if you'd like. Yeah?
Any other questions? Thank you, Dr. Rodriguez, for your presentation.
Thank you.
We will now present Dr. Shauna Manning. She will also be online. So we're ready to put her on the screen. Meanwhile, Dr. Manning is a phycologist working at the intersection of molecular biology and analytical chemistry, whose research examines the dynamics of harmful alga blooms and proliferations. She is also interested in the immense potential of algae in biotechnology for carbon capture, remediation, and the synthesis of diverse bioproducts. Outside of the classroom, Dr. Manning oversees the water omics facility for the Institute of Environment of the Florida International University. She is committed to the advancement of algae science and works closely with colleagues across the globe in academia, local and federal agencies, and also nonprofit organizations. Dr. Manning serves on numerous editorial boards and contributes to the international scientific community through her service to the Phycology Society of America and Algae Biomass Organization, including the development of educational curriculum for the Algae Foundation.
I'd like to. Excellent, thank you. and thank you for accommodating me online. Thank you so much for this invitation. As Alak knows, I would normally be talking about doom and bloom with algae, but today I'll be talking about the potential for algae and the phytochemicals that they're making. So I've entitled this talk Algae Farm, so not your traditional farm, but you've seen a lot of algae today. And we can't talk about the algae without talking about the immense biochemical and genetic diversity. The heartbeat of photosynthesis starts down here with the cyanobacteria, and through numerous endosomatic events led to over a dozen different discrete groups of algae with discrete products. And if you think about land plants being very diverse with all the floral diversity, or all of the animals, you, me, all the fishes in the sea and insects, algae have more genetic and biochemical diversity than all of the zoos and botanical gardens in the world combined. So algae has really become an area of interdisciplinary research. You might be a traditional phycologist working here in the realm of basic biology. You might be out in the field collecting field specimens. Or you might be looking at how algae can bioremediate wastewaters, CO2 emissions, or the potential for making new products. And here we have a new field called algaculture. And here are some small-scale ponds out in Arizona where they're actually growing algae for a purpose. We all know that algae are photosynthetic machines. We know that algae can thrive in a much broader range than land plants and can be grown in non-arable land, not competing with agriculture. They're much more efficient in their energy conversion, usually because they're small size, high surface area to volume ratios. They can absorb nutrients and grow very rapidly, and they're producing way more biodiverse chemicals than plants. So we should think of every cell as an energy factory that is growing and outpacing all of our terrestrial, taller, drier algae, and then we're going to get a lot of interesting chemicals out of our algae. So we tend to think of this as applied phycology, using algae for a purpose, really focusing on the central dogma of producing biomass. You can use a whole biomass, or you can break it down into its constituent parts like proteins, lipids, carbohydrates. People have made topical applications for fertilizers. There are nutraceuticals on the market now. There are specialty chemicals being explored. And of course, fuels have been something that have been long explored with algae, with algal biofuels. So if you talk to any student, the first thing when you ask them about algae, you're usually going to go, ooh, ick. They go, ah, biofuels. And this is an example of Botryococcus bronnii, probably one of the most oleaginous algae we have out there in nature. It is such a slow grower. And so we're really trying to get to the goals of biofuels that meet market prices. But as of right now, we just do not have the price point with growing algae, harvesting algae, and isolating products to get to that price point. But we have algae products and food things all over in the grocery store. So if you go out to eat sushi, you've been exposed to algae, even in your ice cream, your Sara Lee cheesecake, your Sunny D, your salad dressings. Many of those types of products will not stay emulsified without their lovely algens. You're going to have jellos and brownies and all kinds of things that have carrageenans. And you even got diatoms in certain types of products like toothpaste and medicines and things for the garden. So essentially diatoms are like glass, and so it prevents insects from crawling closer to your vegetables. Now we tend to think of algae products in two different categories. We have these commodity algal products that are made in mass for a low price point. Biofuels, as I mentioned, we're trying to get there. But we already have algal biofertilizers and different feedstocks. So if you go to the pet store, you're going to find algal-based fish products. You're going to find fertilizers that will make your tomatoes bigger and hardier. But we also want to think about higher value algal products, things like omega-3s. These are things that algae make and we have krill take it up. So usually people are taking their krill oil, which can have a really fishy aftertaste. So we're taking out the middle fish and going for the source, which is the algae. You can also get lots of different carotenoids, like astaxanthin, beta-carotene, and we're also starting to develop other types of compounds like sulfated polysaccharides, which I'll talk about a case study, and other bioactives that are commanding much higher price points than biofuels. So if you want to go after the $1.50 per liter products in the biofuels realm, or if you want to go after the $6.5 billion kilogram product, we can talk about algal natural products. So here's an overview of algal metabolism. So just a little bit of CO2 and water to make sugar, and then the cascade of metabolism begins. I want you to focus here primarily on secondary metabolism. So secondary metabolism are compounds that algae make to defend themselves. Sometimes they're toxins, sometimes they're protective pigments, but usually in the shikimate pathway, acetyl-CoA pathway, mevalonate pathways, and the non-mevalonate pathway in the chloroplast, also synonymous with the cyanobacteria, we can get a lot of diverse compounds. So we get lots of different types of bioactive compounds coming from microalgae and cyanobacteria. These are polyketides, non-ribosomal peptides, alkaloids, and different polysaccharides. And all of these different compounds tend to have some type of impact, whether it's cytotoxicity, hepatotoxicity, hemotoxicity. So all types of toxicity, keep that in mind because we're just talking about bioactivity. So when we think about these types of compounds, they do closely resemble other types of pharmaceuticals and therapeutics, including chemotherapy agents. So just looking here at Taxol, that was the first chemotherapy agent isolated from a plant. And then we've got a whole host of different types of antibiotics coming from different sources. And really the difference, if you were to overdose on any of these compounds, they would be toxic. It's really about dosage and delivery of any of these types of therapeutics. And we're predicting thousands of different new metabolites coming from algae. So I'll just kind of move into a short case study in my laboratory that's looking at sulfated polysaccharides of porphyridium. So here, this is a set of cultures. These are brackish to marine porphyridium, so they appear kind of pink red. And by contrast, these are also genetically porphyridium, but they are freshwater, and they look blue-green. They're just not expressing the phycoerythrin compound that they do in the marine setting. Zooming in, we can see the cells are quite small, about 5 to 6 microns across. And these cells are very interesting because the cell has a modified cell wall that is made of sulfated polysaccharides. And it kind of works as a concentric ring pushing out from the cell surface, creating these layers as it extends into the supernatant, as they stretch and then break off and then slough into the surrounding medium. And so we're looking primarily at the material that is being sloughed into the medium, which is a sulfated polysaccharide. So this is an example structure of a sulfated polysaccharide. It's composed of varying sugars, could be pentoses and hexoses, and varying degrees of sulfation. So first, we wanted to look at porphyridium and how it grew under different dosages of magnesium sulfate heptahydrate, which is one of the primary constituents to make sulfated polysaccharides. And we can see under the varying concentrations, they really do grow quite similarly to one another. And we do start to see some interesting trends up here towards the top as the cell cultures become more dense with more cells per mil. They're also becoming more viscous as more SPS is being shed off into the medium. So we did identify that cultures A and B were the best performers for the at least growth of porphyridium under these conditions. And then we isolated the extracellular sulfated polysaccharides, looking at both the low molecular weight SPS, that's under 500 kilodaltons, and then we look at all the high molecular weight over 500 kilodaltons, and we measured at 2 different time points at 7 days and at 14 days under the different magnesium sulfate heptahydrate concentrations. And for the most part, there's a linear curve, a linear trend with regard to dosage of magnesium sulfate heptahydrate, with the highest amount of low molecular weight being produced in both 7 and 14 days under 80 millimolar conditions. However, it didn't seem to contribute too much to additional high molecular weight when compared to the control conditions, which is 5 millimolar. Either way, this does give us some insights in how porphyridium grows, its productivity, and if we want to isolate either low molecular weight or high molecular weight, the conditions that we would grow it under. We took it a little bit further and did some LCMS-MS to kind of look at the composition of this SPS. And we saw that generally it was about 51% pentose, about 43.3 hexose, and we had some deoxyhexose, uronic hexoses, and these amino acetylated hexoses. And then so we wanted to test these particular fractions on actively growing bacterial cultures, as we tested both gram-positive and gram-negative pathogenic bacteria. So as the bacteria were entering logarithmic phase, they were dosaged with either a control, an antibiotic, or different concentrations of sulfated polysaccharide. Interestingly enough, we can see that upon application here with Staphylococcus, it does arrest growth. So we do have a minimal inhibitory concentration of 10 micrograms per mil here. Interestingly enough, the tetracycline did recover, suggesting antibiotic resistance. And this is the primary thing we're really working around, is getting around that antibiotic resistance in traditional antibiotics. When looking at Pseudomonas aeruginosa, we do see that SPS did arrest growth, and we had dosages of 500 and 1,000 micrograms per mil. So it did take a bit more to kind of halt Pseudomonas from growing. With Acinetobacter baumannii, we did have treatments of 10 micrograms per mil that halted growth, although we did see some recovery of those cultures after some hours, although under a higher dosage, it did arrest. Although we did see, again, with traditional types of antibiotics, we did have carbonicillin resistance as the carbonicillin cultures continued to grow after a few hours. And lastly, with Bacillus subtilis, we did see we had a minimum inhibitory concentration at 10 micrograms per mil. So all of these give us a lot of hope that the use of SPS could be used towards pathogenic bacteria. And so these are actively growing bacteria. The other thing we wanted to be able to test are to prevent the growth of bacteria. So using plates and having plates that do not have SPS infused on the left versus SPS infused plates on the right, we can see that streaking Staphylococcus. On the control plates, we have more colonies after 24 hours and 48 hours than we do with our treated samples. We're going to continue on with additional types of antibiotics, looking at zones of inhibition. So please stay tuned for new news on the Neosporin. And to segue out of that, just talking about yet another bioactive that's really, really popular is astaxanthin. I took this facility from 15 milliliters of culture to 360,000 liters of growing algae for the production of astaxanthin, a really potent carotenoid antioxidant. I've worked with the country of Qatar on making algal feedstocks. It's so hard in a desert country getting good agricultural silage, and algae has proven to be a really good replacement. It's even improved the health of the animals as well as the products of the eggs and the milk and the meat. And then lastly, we can actually extract some of the oil for an energy product, like for the Department of Energy, and have a carbohydrate protein-rich product that we've used for feed. And we actually compared feeding a defatted versus fully fatted algal feed to chickens and showed that actually improved the health, and there was no impact on the chicks. In fact, it improved their health. So when we think about algal culture, We get them all the traditional compounds that can come out of here, or really the sky's the limit. So I just want to thank you. I would like to thank those in the main lab that helped out with some of the research that I showed you here today. Thank you to ALOC for the invitation, and thank you for having me. I'll welcome any questions.
Do we have any questions? Yes, we have one question.
Hello? Great.
I'm so sorry, my camera's not working. Please ask.
Hi, thank you. Thank you for the talk. It was very interesting. I have a question. We talked a lot about culture of algae. Do you see, you can culture very specific strains to produce very specific products. Do you see, how does that intersect with harvesting, say algal blooms? Is there any application for the use of algae in that situation where we have huge algal growth for excess nutrients, et cetera? Do you see there kind of any application there?
Absolutely. Oh yeah, no, there have actually been some pretty, creative applications. So if you think back to the 2008 Qingdao event where we had that ulva bloom that just kind of overwhelmed the coastline of the Yellow Sea, that particular material was harvested. It was made into biofertilizer, biochar, silage for livestock. So, that's more of a nuisance bloom. If it did degrade, you would have huge plumes of hydrogen sulfide, so still not good either way. But if you think about, Lake Erie, Lake Okeechobee, Lake Klamath, where we have these huge inundations of microcystis, these very, thick, scummy cyanobacterial blooms, there are some technologies that they're utilizing right now. They have these boat harvesters that are essentially going in and they run that belt backwards, so it's just literally lifting it off of the surface of the water and it's collecting it into bins. Now, that doesn't, give us an end product, but there are applications. So a colleague of mine has taken some of that quote unquote called waste biomass, conditioned it into a high protein foam. So essentially he's utilizing these foams as bio replacements for traditional insoles, midsoles, yoga mats, anywhere we need some type of foam polymer replacement where we're not generating a new plastic. So there are these new applications. I'll mention one other is utilizing, some of these different compounds from algae for making flip-flops and surfboard foam. So we are thinking of utility of this waste biomass. It's just that one, containment, two, care, especially around, the toxic material and exposure, and then, getting it into a usable form so it's, something functional and not toxic any longer.
Thank you very much. Please, another round of applause. Thank you, Dr. Manning. I would like to invite Dr. Natasha De Leon, President of the American Association for the Advancement of Science, Caribbean Division, who will bring us some information about the association.
Gracias, ¿se escucha bien? Sí. Nada, voy a hablar un poquito en español, then I will switch to English, and I will be doing this crazy Spanglish presentation. Pero gracias, first of all, thank ALOC for letting me have a few words and explain to you what AAAS Caribbean Division is and LSCN. Para los que saben qué es AAAS es la organización más grande, yo creo, de ciencia que hay en la nación. También son los que se encargan de publicar la revista Science, que algunos la han visto por ahí, ¿verdad? Y todos queremos publicar ahí en algún momento de nuestras vidas como científicos. Así que nosotros aquí en el Caribe llevamos 38 años con la división. Existen 3 divisiones a nivel de Estados Unidos. Una de ellas, ¿verdad?, está aquí en el Caribe y empezó en Puerto Rico, en el recinto de Mayagüez, y se ha esparcido alrededor de toda la isla. Nosotros cada año hacemos una convención anual ¿verdad?, en la época entre octubre, noviembre, después de los huracanes. Así que siempre tenemos nuestra convención anual para que estén pendientes a esa fecha. La convención siempre trata de llevar alguna temática que sea relacionada a problemas de política pública y problemas, ¿verdad?, que estén en el país actualmente. Contamos con cientos de participantes alrededor, ¿verdad?, de toda la isla. Al momento hemos tenido, entiendo que creo que se me está escapando otro fellow del área de comunicaciones, o que tenemos un Communication Fellow. Hace tiempito ya fue el doctor Kevin Alicea, que está ahora mismo en Macao. Y ahora tenemos un LSCN, que les voy a hablar un poquito de qué es este programa, un LSCN Liaison, que esa sería yo. Estos son un poquito de fotos sobre la última convención que le hicimos en Casa Pueblo, que se trataba de ciencia desde la comunidad. Así que nos dirigimos a Casa Pueblo, que es una organización comunitaria basada en ciencia, y estos son el poster board que tuvimos, ¿verdad?, el poster session, donde fue en la Galería de Arte. Así que tuvimos un poquito de ciencia, comunidad y cultura. ¿Qué nosotros hacemos en AAAS Caribbean Division aquí en Puerto Rico? Pues tratamos de llevar algunos eventos a través del año, no solamente la convención anual. Hacemos lo que se llaman los Science Café, que buscan, ¿verdad?, crear o aumentar ese trust de las comunidades con los científicos. Así que nosotros llevamos temas que sean de interés público, llevamos un científico o personas que sean del área para que hablen con la comunidad en un ambiente que sea en la comunidad, ¿verdad? Decimos que son Science Café, pero muchos de ellos terminan siendo en alguna barra, pues por donde llevamos, ¿verdad? Y nos llegan los estudiantes, los profesores y los miembros de la comunidad. Así que no solamente vamos a convención, hacemos conferencias, pero tratamos de llevar esa ciencia relevante a las personas y al público general, a quien, ¿verdad?, nosotros como científicos le debemos estar sirviendo. Así que estos son algunos ejemplos de temas que hemos estado trabajando. Y entre ellos, un día tuvimos uno que fue sobre el microbioma humano y la nutrición, y lo hicimos en Lote 23. Tuvimos uno sobre alcoholismo y adicciones en, ¿cómo se llama este sitio? En Carolina. Vigo Beach Club, donde hacen Ocean. Así que hemos tenido, ¿verdad?, todo este tipo de eventos que van a volver a comenzar luego de ¿verdad?, ya terminamos la pandemia, así que estamos volviendo a los eventos presenciales. Así que pendiente al calendario, que van a estar surgiendo unos cuantos este año. También tratamos de crear ese liderato alrededor de la ciencia en Puerto Rico, también, ¿verdad?, en ese gol de seguir aumentando esa confianza, y contamos con colaboración con diversas organizaciones sin fines de lucro y universidades. Nuestra junta de directores tiene personas, ¿verdad?, de Intel, UPR, el Puerto Rico Science Trust, y colaboramos con organizaciones, desde organizaciones para jóvenes como Mentes Puertorriqueñas en Acción, el Puerto Rico LSAM, la Asociación de Química y Casa Pueblo y Caminos en Ciencia, que es un podcast que les recomiendo que sigan. Entonces, LSCN, antes de que comience el video, LSCN es el nuevo programa de la AAAS que busca aumentar y crear una red local de participación. El plan de esta organización es aumentar, como les mencioné, la confianza del ciudadano, de nuevo, en los científicos y en la ciencia que se crea. Llevar la ciencia nuevamente a las comunidades y a los científicos a buscar, ¿verdad?, esos problemas que tiene nuestra comunidad y trabajar towards that. Les voy a poner este videito que creó el proyecto para Puerto Rico. Así que esto fue cuando lanzamos LSCN ahora en noviembre. Así que si pueden ahora sí ponerlo un momentito. Tiene volumen.
Trabajo para la Asociación Americana para el Avance de la Ciencia, la cual es conocida también como AAAS por sus siglas en inglés. Y hoy tengo el placer de hablarles un poco sobre la Red Local de Participación Científica, conocida también como LSEN por sus siglas en inglés, y a la vez contarles por qué nos alegra tanto que Puerto Rico ya sea parte de este esfuerzo. La Asociación Americana para el Avance de la Ciencia está comprometida a generar vínculos de confianza entre científicos y líderes cívicos, y LSEN es nuestra iniciativa emblemática, la cual busca impulsar el progreso hacia este objetivo por medio de la movilización de profesionales en las áreas de ciencia, tecnología, ingeniería y matemáticas, a través de redes locales y regionales en todo Estados Unidos, y así interactuar de una manera aún más efectiva con sus comunidades. Pero no podemos lograr esta visión sin profesionales en dichas áreas dispuestos a liderar estos esfuerzos, comenzando en sus propias comunidades. Es por eso que este año AAAS ha impulsado la creación de los primeros 6 centros locales de la Red de Participación Científica Local, LSEN, abriendo así un espacio en donde los profesionales en las áreas de ciencia, tecnología, ingeniería, matemáticas y entusiastas de la ciencia puedan reunirse y participar. En cada una de estas redes, LSEN trabaja y trabajará con líderes científicos locales como la doctora Natasha de León Rodríguez y el doctor Kevin Alecea Torres para fomentar, equipar y activar profesionales locales en las áreas de ciencia, tecnología, ingeniería y matemáticas para que trabajen dentro de sus propias comunidades. Proporcionar oportunidades para el desarrollo y perfeccionamiento de habilidades a través de actividades participativas. Construir redes que fomenten el aprendizaje y la colaboración entre pares, así como la creación de más redes. AAAS está muy agradecida con la doctora Natasha de León Rodríguez y el doctor Kevin Alecea Torres por su entusiasmo y liderazgo. Para nosotros es un orgullo que sean parte de nuestro equipo y los invitamos a todos ustedes a que se unan a la red que ellos están creando aquí en Puerto Rico. Algunos de los beneficios de unirse a la red de LSEN son oportunidades de desarrollo personal y profesional, acceso a formación y recursos especializados, visibilidad y reconocimiento profesional, colaborar en el proceso de generar confianza y visibilidad en el uso de la ciencia, ser parte activa en el proceso de mejora de estados y localidades que necesitan más acceso a la ciencia. Unirse a la red LSEN en Puerto Rico es gratis. Si usted ya tiene una cuenta de usuario en AAAS, lo único que tiene que hacer es entrar con la misma contraseña en el sitio web que se muestra en la pantalla. Pero si usted todavía no tiene una contraseña, el primer paso es seguir las instrucciones para crear una cuenta de usuario en la página de inicio. La realidad es que las personas necesitan la ciencia y la ciencia necesita personas. Únase usted hoy mismo a la red de LSEN y así juntos podremos continuar trabajando en generar confianza en la ciencia y fomentar aún más la participación cívica a nivel local.
Como pueden ver, el LSEN va a estar comenzando a tener sus actividades este año. Ahora pueden escanear el QR code para inscribirse en el mailing list, donde le vamos a estar enviando información sobre las actividades. Vamos a estar haciendo un poquito de touring alrededor de las universidades y las organizaciones para comenzar a expandir la red. Y van a haber varios foros que van a ser virtuales, así se pueden conectar desde cualquier parte. Algunos foros van a ser, ¿verdad?, creados desde Puerto Rico para las personas de habla hispana, otros van a ser en inglés desde el LSEN a nivel nacional, pero sí están abiertos para toda la red para que se puedan unir. La dirección que salió anteriormente es un foro donde esperamos participación de las personas que puedan conectar con otros científicos, ya sea locales, de la diáspora, etcétera, y que puedan ampliar ese conocimiento, ¿verdad?, de participación ciudadana. Algo que es importante, AAAS, aunque nosotros, ¿verdad?, estamos enfocados muchas veces en STEM, también tenemos y está abierto para todo lo que es ciencia, desde ciencias políticas, economía, sociales, matemática, todo, ¿verdad? Todo lo que es ciencia, investigación y cree conocimiento es parte de lo que es la Asociación Americana para el Avance de la Ciencia. Así que los invito a formar parte de la red. Próximamente estaré enviando un email con las formas para que se puedan unir. Como mencionamos, no necesitan ser miembros activos de AAAS, ya que eso, ¿verdad?, tiene un costo, así que conectarse con la red de LSEN es completamente gratis. Dudas o preguntas me pueden encontrar por ahí por los pasillos antes, cuando se acabe el evento. Pero sin más, gracias por darme espacio para estas palabras. AAAS también aquí en el Caribe quiere reconocer la labor que han hecho muchos científicos que han pasado por aquí el día de hoy en su trabajo, ¿verdad?, y contribuciones a lo que es la agricultura en el Caribe. So with that, AAAS would like to recognize the amazing contribution, outstanding service contributions in the field of agriculture and here in the Caribbean. So we're going to give some recognitions to some folks that are here present. So we would like to recognize Dr. Sonia Peters for her outstanding service in the field of biocultural education. And we are going to have some pictures of posts. We can have picture in the middle. In the middle. In the middle? Okay. Yeah. Yeah. There you go. Thank you. Thank you for your. I'm going to move. I'm moving here. Also, we'd like to recognize Dr. James Giovanni for her outstanding service and contribution in the field of plant genomics. Thank you. I think we need to over here. Thank you. Okay, thank you. Also, Dr. Thierry Hans for her contribution his contribution in the field of entomology and crop protection. Thank you. Thanks. Thanks. Now, one of our own, Dr. Juan Negrón Berríos, for his contribution in the field of biotechnology and education, biotechnology education in Puerto Rico. Dr. Diane Beckles for her outstanding service in the field of post-harvest integrative biology. Thank you. Thank you. And Dr. Alok Aron for his service in mentoring STEM students from Puerto Rico. Thank you. And with that, I don't know if we're closing, but you will close probably all this ceremony. But thank you so much for the opportunity, and I hope you actually enjoy lunch that was sponsored by AAAS. So thank you for having me, and have a good night. Good day. Nice day. I'm ready to speak.
Thank you, Dr. De Leon. Today, indeed, we encounter interesting, valuable, and significant information to learn and also to implement, not in Puerto Rico, also in the Caribbean and the world, because we need it. So we express our gratitude to all the speakers for accepting our invitation. The quality of each presentation is an indicator that we really met the objectives that we wanted to for this activity. So thank you very much. We appreciate your collaboration. Before we conclude, do not forget to complete the evaluation for this activity. The QR code is in the screen. For those on the web, the QR code will be written in the chat. We would like to thank the United States Department of Agriculture, Corteva, and the American Association for the Advancement of Science Caribbean Division for sponsoring this activity. We also want to thank Dr. Juana Negrón Berríos, Dr. Alok Arun, and all of our guests and their participation. A special thanks to all the collaborators of the Barranquitas campus for the support given to this activity. We hope that you have enjoyed the symposium. Thank you once again. See you next time.